Hello all, welcome to the session on neural excitability, synapses, and glia. My name is Kirti Krishnan. I'm an assistant professor at the University of Tennessee in Knoxville. My research interests are mouse models of neurodevelopmental disorders with a focus on cellular and behavioral neuroscience. I'm excited to be your MC today, along with William Chapman, who is going to be our backend host. I'm really looking forward to exciting cellular neuroscience today. So please ask questions in the Q&A and post chats to stimulate relevant discussions. Panelists are welcome to do this as well. Since we have to abide by time restrictions, I would like to request the speakers to please keep to your allotted time. So 10 minutes-ish, I'll show you a card. I'll hold up signs, and then we'll try to wrap things up. So with that basic introduction, I am so glad to introduce our first speaker, Najee, from the Institute of Psychiatry and Neurosciences of Paris in the lab directed by Dr. Maria Cecilia Angolu. Their lab is interested in the interactions between neurons and oligodendroglia in the myelination process and myelin repair. Her title is Myelination of Parvalbumin Interneuron Shapes the Function of Cortical Sensory Inhibitory Circuits and Mouse Whisker-Mediated Behavior. Welcome, Najee.
Thank you, Kirti. Good morning, everybody. So today I'm going to talk about one of our work, which is about the role of myelination of parvalbumin interneurons in the function of cortical sensory inhibitory circuits and mouse whisker-mediated behavior. So OPCs are the precursors of myelinating oligodendrocytes, which principal function is neuronal myelination that is important to facilitate fast alternatory conduction of action potential. And several teams, including our, were involved in the characterization of the first non-neuronal synapse between OPC and axon. And we showed in a published work.
Sorry, Najee,
yeah.
I don't see your screen right now.
You don't see my screen? Okay.
Are you sharing your slides?
I guess, sorry. Yeah. Now?
Yes, thank you.
Okay, okay, sorry. So yeah, and so I was saying that we showed that regarding GABAergic synapses in OPCs, that fast-spiking interneurons are highly connected to OPCs at postsynaptic sites containing the GABA receptor, including the gamma-2 isoform, while non-fast-spiking interneurons are poorly connected to OPCs through postsynaptic sites lacking this isoform subunit. So as I just said, OPC receive a large GABAergic synaptic input from fast-spiking interneuron containing the gamma-2 GABA receptor. And it's not known whether the synapse could impact myelination of those interneurons. And interestingly, several studies revealed that a large fraction of neocortical myelin insheets axon of fast-spiking interneuron. So we found interesting to evaluate whether fast-spiking interneuron OPC synapses could guide the correct myelination of fast-spiking interneurons. So our strategy was to generate a mice model characterized by a loss of function of the gamma-2 subunit in OPCs. So we use a NG2 creux G-country flux gamma-2 flux mice that induce a loss of expression of gamma-2 in OPC, and this will generate a dysfunction of the gamma-2 mediated synapses. And we use the NG2 creux G-country flux mice as a control. So to compare the myelination of fast-spiking interneuron in our two model, we loaded with biocytin fast-spiking interneuron from the barrel cortex, the layer 4 of the barrel cortex. And so here you can see reconstruction and representative confocal images of fast-spiking interneuron with biocytin in red and myelin in green. And so we observe that all fast-spiking interneurons are myelinated in both groups. And then we looked more specifically to the myelin distribution of fast-spiking interneuron. And so we observed that the distance from the soma to the onset of MVP is increased in the mutant mice compared to the control, as you can see here. And this is associated to an increase of the proximal segments in the mutants, so as you can see here. And we also observed that the mean internode length per cell is increased in the gamma-2 flux mice compared to the control. So we can conclude that the disruption of fast-spiking interneuron OPC synapses cause myelination defects in fast-spiking interneuron. In addition to internode, we also looked at the node of Ranvier in our two models, since it's known that nodes of Ranvier are important in the optimization of action potential propagation and also in the conduction velocity. So here you have confocal images of node of Ranvier in control and in the mutant mice. And node of Ranvier were identified as the structure between two consecutive internodes flanked by Casper-labeled paranodes. And so we found that the mean node lengths per cell were increased in our mutants compared to the control. And this is associated to a shift of the distribution of the node lengths toward higher size. So we can say that the disruption of these synapses also cause node of Ranvier defects in fast-spiking interneuron. So to evaluate whether those myelination defect of fast-spiking interneuron could be due to morphology alteration of oligodendrocytes, we also studied the morphology of those cells. So we identified oligodendrocytes with their linear IV curve and also with specific markers. And so we did morphometric measurements using shoal analysis, and we found that the number of intersection of oligodendrocyte from the mutant compared to the control were reduced. And we also observed that the number of processes from the soma was also decreased in the mutant compared to the control. So we can conclude that differentiating OL show a lower level of ramification in the gamma-2 mice compared to the control. So then it's well known that myelination distribution and also axonal structure are involved in action potential discharges. So we wanted to evaluate whether the modification of myelination observed in fast-spiking interneuron could affect the electrical properties of those cells. And so we did patch clamp, and here you can see representative action potential discharges of fast-spiking interneuron in control and in the mutants. And we found a decrease in the firing frequency of fast-spiking interneuron in the mutants. So we can say that fast-spiking interneuron myelination defects compromise their high firing frequency. We also wanted to evaluate whether fast-spiking interneuron myelination defect could impair the function of fast-spiking interneuron in inhibitory circuits by studying the thalamocortical feed-forward inhibition, since fast-spiking interneuron are important to control the balance between excitation and inhibition in the network through feed-forward inhibition, which is process that allow direct thalamocortical excitation of fast-spiking interneuron and excitatory cells. And in turn, fast-spiking cells will inhibit the excitatory cells to avoid recurrent excitation and maintain a dynamic balance between excitation and inhibition that is important in the processing of neocortical information. So basically what we did, we used somatosensory thalamocortical slices, and we were able to stimulate electrically thalamic input while recording inhibitory and excitatory postsynaptic currents in the spiny stellate cells. And so here you can see representative currents observed in control and in the mutant mice. And what we observed is that the excitatory postsynaptic current was similar in our both group, showing that the excitatory thalamocortical inputs are not affected in the mutants, while we found that the inhibitory postsynaptic current was significantly decreased in the mutant mice compared to the control. And this generates an increase in the ratio between excitation and inhibition in the mutants, generating a strong imbalance in the network. We also quantified the latencies of excitatory and inhibitory postsynaptic currents. And while we observed that the latency of EPSC was unchanged between both groups, we observed an increase in the iPSC latency in the mutant compared to the control. We also quantified the predicted conduction velocity for fast-spiking interneuron according to the size of the internode and the node of Ranvier that we observed in our groups. And we found that this conduction velocity was reduced in the mutant compared to the control. So we can say that abnormal fast-spiking interneuron myelination is associated with impaired fast-spiking interneuron inhibition of spiny stellate cells, probably due to a deficit in fast-spiking interneuron action potential conduction. So then we wanted to evaluate whether fast-spiking interneuron myelination defects and also the dysfunction of the inhibitory circuit that we observed in the barrel cortex could induce impairments of the whisker sensory behavior. And so we subjected mice from the control and gamma-2 group to a whisker-dependent texture discrimination task. So this task is composed of 3 phases. That is based on the ability of the mice to discriminate between objects with their whiskers. So the first phase is an habituation phase to promote the exploratory behavior. The second phase is a learning phase where the animal is subjected to 2 similar texture objects. And in the third phase, the testing phase, a new texture object is added to the environment, and the animal is supposed to discriminate between the old object and the novel object by spending more time exploring the novel objects. So what we do found is that during the learning phase, the whisker-based texture exploration was the same between the both group. Showing that animals from both groups have similar capacity of exploration. And we also found that control animals are able to discriminate between the 2 objects since they spend more time exploring the novel texture object compared to the old one, while the mutant was not able to discriminate between both objects since they spend similar time exploring the novel and the old one. So we can say that abnormal fast-spiking interneuron myelination and function induce deficient whisker-dependent texture discrimination in gamma-2 mice. So to conclude, this work showed that myelination of parvalbumin-positive fast-spiking interneuron in the barrel cortex is critical to the morphology of the proximal axon and the high firing frequency of fast-spiking interneuron. Also, it's important to the function of fast-spiking interneuron-mediated inhibitory circuits and to the control of whisker-mediated behavior. So to finish, I would like to thank all the members of my team and more specifically our PI, Maria Cecilia Angulo, and also Marie Vidal and Madalena Balia, who were directly involved in these projects. Thank you very much. And if you have any question, I would be happy to answer. Thank you.
Thank you, Najee. We are waiting for questions from the other groups at this point. If the panelists have questions, they're welcome to start, or else I have a few questions as well. So Bradley, would you have any for Najee?
I can't think of anything right now.
And we seem to be missing our second speaker at this point. So I think we're going to try to do a little bit of more of a discussion with your topic, and then perhaps we could start, Bradley, a little bit earlier, but maybe not too much because I don't want other people to miss the talk if they were planning on it. So my question, and I might have missed this a little bit, I was curious about the knockout animal here, the gamma-2. Is it a full null? Can you talk a little bit more about the knockout itself?
So the model, so it's a specific, so it's an induced with tamoxifen model mice, and so the deletion of the gamma-2 subunits occurs specifically in NG2 cells, and so in OPC. So it's really specific of OPC, and the idea here with this mutant was to induce a dysfunction of the synapse, specifically in OPC, to be able to study the gamma-2 mediated synapses between interneuron and OPC cells.
Okay, great. Yeah, that's.
Does
exactly.
it answer, yeah, to your question?
Yes, thank you. So do you have a timeline of when does it normally start showing up? When does myelination normally start showing up in the paravalbumin cells? I saw data from your P30. Is that the time period that it starts at, or have you done a time course analysis for this?
Yeah, so we look at P30 because we know that the circuit that we studied are quite mature at this age. So the myelination in mice, I would say, is considered as mature around 2 months.
Okay.
So, but I think that at P30, it's already in an advanced stage. But we looked at this age to be to stick to the circuits, in fact. But we also looked at the global myelination later. But if we looked at P30, it's more because of the circuits.
I see. Okay. And your behaviors that you're performing for the novel object recognition with the whiskers, what age are you doing those?
It was done at 3 months.
3 months. Okay, I see.
Yeah.
Okay, great. Yeah. The other questions I had were more related to the areas themselves. I find paravalbumin neurons fascinating. That's my subject of research as well.
Yeah.
So and I've noticed regional distributions, right? So between visual cortex versus motor versus somatosensory. Do you think the myelination is a specific feature of somatosensory PV neurons, or do you think this is more global to all PV cells in the brain?
So what we do know about myelination of PV cells, and what I found quite interesting, is that they represent mostly all the interneuron myelinated cells, the PV cells. So the other type of interneuron, like somatostatin, have very few myelin. So then I would say, regarding of the organization of the myelin in these cells, it should be quite similar in the different area that you are talking about. But what is really specific of those cells is that they really represent most of the myelin of inhibitory axon. And that's why we were quite interested in the organization of the myelin of fast-packing interneuron, because if we go back to the synapse, we also observed that OPCs are highly connected to fast-spiking interneuron. And so the fact that they also represent most of the myelinating inhibitory axon, we found interesting to study if it could have a link between the synapse and also the myelin of those cells. Then we focused more specifically on the somatosensory, because we studied the synapse in this area. Now we are also looking in the prefrontal cortex, because we would like to look a little bit more to the cognitive process. So we know that PV also regroup most of the myelin in this area regarding interneuron. And yeah.
And are you also specifically looking in the layer 4? So I see your data was from the layer 4, but do you think it's the same between the different layers, or is there something special about the, gating the thalamocortical input side of it that makes the PV neurons in layer 4 so more special to this aspect?
Yeah. So regarding the myelin, so the defects that we found in layer 4 in our model are also observed in layer 5. So we started first in the deeper layer because we know that PV cells are quite important in this area, in this layer, sorry, in the deeper layer. And then it's true that we focused more on the layer 4 because we wanted to look at a circuit that involved specifically the fast spiking interneurone. But what I can say is that in deeper layer, in layer 5, we also see those defects in the mutants.
I see. And so could you speculate a little bit about what do you think the consequences are from the deeper layers, like layer-specific function of PV neurons and their myelination versus, right, so layer 4 versus in terms of gating and perhaps as far as the output of these neurons as well?
Yes, so in fact, the interest of this work was more to try to understand if this synapse could modulate the myelin of fast spiking interneuron and also their integration in specific circuits. So regarding the layer 5, we found also similar defects. We didn't go that far in the analysis, but we know that myelin distribution is affected. So I would say that probably this synapse is important to control the distribution in the myelin, to control the conduction velocity of those cells, because they are really characterized by their high firing frequency. And then it must be also important to the connection between layer, probably. So we didn't look at that, but it's probably important. And we also observed that in layer 5, the firing frequency is reduced when we have the defects, also like in the layer 4.
Okay. So any questions from any of the audience? Oh, we're getting one. Great. I do have more questions for you, but I'll start with this one. Shiona says, I'm sorry I missed most of your talk. We'll definitely look at the recording later. This may just be a tangential question. Do we know if fast spiking interneurons in the striatum are similarly myelinated?
In the striatum, I'm not sure that any study showed that. It was shown in the somatosensory cortex, prefrontal cortex, also in the hippocamp. If the question is just about knowing if interneurons are myelinated, I mean PV interneurons are myelinated in the striatum,
Did we lose Najee?
I believe we did.
Oh, okay. Well, hopefully she will collect back and get back to answering her questions. We have a few minutes while we are going to wait here. Bradley, are you still with us?
Yes, I'm here.
All right. Okay. William, would you be able to get her screen off by any chance? Because Bradley won't be able to do that, right?
Yep, I can do that.
Okay. We might just have to.
I'll reach out to her. If you're watching, maybe, shoot us an email if you're having technical issues. Right. So this morning is an interesting one. That's all I can say. We're missing our second speaker, and I'm going to spend a little bit of time, give William a chance to reconnect over here, and Bradley can perhaps get ready to get started on this next talk.
Okay.
I really didn't want to go too early, and if people were going to miss your talk and you were 5 minutes ahead of schedule, that might throw some people off. But it'll give you more time to talk about your research. So that's the good news from that side, right? So we could have more of a discussion. So from that point of view, I am glad to introduce our second speaker, Bradley Alyzer, who is a senior contributor at the Open Worm Foundation, and is the head of the Orthogonal Research and Education Laboratory. His title is the Psychophysics of Non-Neuronal Cognition. Welcome, Bradley.
Thank you. I'll put a related work in the chat. case you want to look into that further. Let me share my screen.
Thank you.
Okay, oops. So this is the psychophysics of non-neuronal cognition, and thank you to the co-authors here who are here, but I'm presenting on their behalf. We're going to talk about an organism called Basilaria. Actually, they're a class of diatom. And so the diatom is, I'll have to show these videos, maybe to give you a sense of what diatoms look like. They're single cell organisms that exist in multicellular colonies. So as you can see in this film, this is in the diatom, specifically Basilaria in the wild, and they move in this way. They have these, we'll talk about how they move, and you can see that they're moving in water like this, and they fold and make all sorts of different conformational changes with respect to their shape. And then this is a single colony on the right, which is, and you're going to see in this example, one cycle of movement of a single colony. And so here you can see this single colony moving back and forth. This is something that's isolated in culture. And you can see how each cell moves against one another and they make these movements. This is just one type of movement. As you could see from the other movie, they make all sorts of different movements and different orientations. But this is just sort of a controlled way of viewing this. So this is, we're going to appeal to something called psychophysics to talk about non-neuronal cognition. And I'll talk about what non-neuronal cognition is in a bit. It's a little bit of a controversial sort of hypothesis about behavior, but we're going to talk about possible psychophysical models. And so psychophysical models, we think of psychophysics as being a human thing. We think of performance, we think of all sorts of things like that, but then you can also observe psychophysics in different animals, non-human animal models. And so, for example, we can look at psychophysics of the mouse. We can look at their visual system. We can characterize performance quantitatively. We can also look at goldfish. And in goldfish, again, they have their visual system that we can attribute certain sort of quantitative laws to that. But can we also characterize diatom behavior and diatom movement in the same way? And that's kind of what I'm going to propose here. And so these citations at the top and the bottom are different psychophysics references, especially with respect to non-human animals. So these are diatoms, and probably most people in neuroscience aren't familiar with diatoms, but they're a very interesting group of organisms. They're an algae, so they exist in water, and they're single cell eukaryotes. So they're not prokaryotes, they're not archaea bacteria, they're eukaryotes, just like us, but they have plant-like characteristics. So they have a chlorophyll element in the cell, and they have a silica cell wall. So their cell walls are rigid, but they also are eukaryotes. And so you can see on the upper right, you have these specific rapid diatom types. So the rapid diatoms, as opposed to some of the other types of diatoms you see over here, are these elongated shapes. They have here are the different attributes of the cell, these girdle bands, they have different valves, and this is basically the shape. And then on the bottom, you can see that they take on a number of different shapes. They almost like little machines or different plastic molds, but these are cells. And they come in rod shapes, flake shapes, and different 3D shapes. But we'll be talking about the rapid diatom today. It's interesting to note that the size is only about 10 to 80 microns. So that's very small for a single cell. And they exist in these colonies. Not all diatoms exist in the colonies, but these do, the ones we'll be talking about today. So we can analyze bacillary movement in culture. And it turns out you can culture diatoms, and this genus Basilaria is one type of diatom that we can actually culture. So you can go out into the wild, into like a aquatic environment, and isolate these different colonies, put them in a culture. The culture conditions aren't too hard to replicate, and then you can observe these under a microscope. So here's a movie again of the single colony movement. And so again, the movement is isolated on a single focal plane, and you can see how they move back and forth. There's this oscillation in the colony. So each of the cells are moving. They're not moving independently. They're sliding against one another. And you can see that there's like a leading edge to this collective behavior, and then the other cells follow. So it follows a sort of accordion-like oscillation. There are also other phases of movement. So this is an idealized movement, but there are a lot of other phases of movement we can characterize. So there's a free sliding movement. There are changes in momentum during the oscillation. And so even in this small movement, you can characterize the different phases. But as you saw from the example of the wild colonies, that they move in different orientations in different ways that are very interesting. We can model this computationally as a series of coupled oscillators, of course, which I'll show you in this talk, but movement can vary due to stimuli. So we'll talk a little bit about the stimulus conditions and how that might affect the movement as well. So I'm going to talk now about non-neuronal cognition. And so it's a somewhat maybe controversial take on behavior and how behavior is executed. So there are a couple of citations that I've included that maybe are about in the last 10 years where they've tried to define non-neuronal cognition. And one definition from Leon in 2015 is that it's the total set of mechanisms and processes that underlie information acquisition, storage, processing, and utilization. So they think about it much like human cognition maybe, but they kind of give it this idea that you can observe this in systems that don't necessarily have neurons or sensory interfaces in the conventional sense. Beluska and Levin also published another paper where they contemplated what a non-neuronal cognition system might look like. And they make the point, one of the points they make is that the extracellular matrix is ideal as a memory medium. So they propose a bunch of different mechanisms for this. One of those is the extracellular matrix. They also suggest that there's a stigmergy that occurs where physical traces that are excreted in the extracellular matrix can be referenced and read into the future. And so one example from diatoms, bacillary in particular, are these mucilage trails. So remember I showed you these single cells sliding against one another. These single cells, they generate these mucilage trails between them. And so this is one of the things that helps them to stick together, but not, also be able to move sort of in concert. And so it's sort of like this viscous type medium in between the cells. And we can use these maybe as a memory matrix. These mucilage trails are secreted polymers. They completely encase the cell and are responsible for adhesion and interactions with the external environment. So let's think back to the sensory world inhabited by diatoms, and there is a great diversity of diatoms, but they're generally found in these different aquatic environments. So you can find them in marine estuaries, you can find them in shallow waters, you can find them, in places where they're sort of pelagic environments where you have a lot of biodiversity, and you just, go in and you can scoop up from the surface and you can isolate these diatoms. And so they're exhibiting characteristics of both an aquatic species, but they're also subject to light and other chemical stimuli. So one of those stimuli is CO2. And so we know from the literature that there's some effects of elevated CO2 on a natural diatom community. So we can observe that CO2 exists in the aquatic environment. These cells can sense CO2 presence of CO2 and concentration of CO2. And the reason it does so is because it affects the growth of the diatoms in their colonies. High levels of CO2, for example, reduces grazing pressure and has an effect on photosynthesis. So it's imperative for the cells to be able to sense CO2 in some way. And of course, they can sense changes in CO2 as well. They can also do things like sense motion, relative motion, and forces acting upon the colonies. So they exist in these water columns, and they're subject to water shear and other types of pressures like that. And so there are a couple of citations that I've included here on different things having to do with nutrients and environmental signals. And one of those signals is that they detect and respond to fluid motion, changes in fluid, the motion of fluid. So shear, osmotic stress, and iron abundance are all things that they sense and can respond to. Finally, they can also respond to light, as we mentioned previously. And this is a citation where they talk about light intensity in particular. And so CO2 and light availability affect diatom physiology. So CO2 and light in this case act in concert, although they don't have to. And so you can test, say in this paper at the top here, Hayden et al., they tested 3 light intensities. And so they found that low light levels affect physiology and distinct physiological traits. They can also localize sources of chemokinetic and chemotactic mobility. So they can sense chemical gradients, and they can use those as ways to find mates and other sorts of pheromone-guided searches. And so there's a fascinating physiology in the cells where they restore their cell size by reproducing. And so I'm not going to get into that today, but I will say that they use these sensory signals for that purpose. So we can go on to talk about a little bit higher level structure here. And one of the things we can talk about are these collective pattern generators. So these are the cells, again, they're moving against a substrate in this figure, but they're actually using these actin filaments and these myosin elements, and they're generating this adhesive mucilage. And they're moving against a substrate using these adhesives and this actin myosin complex. But imagine that this substrate is another cell, and you basically have a picture of how the Basilaria colonies move. And so these actin and myosin systems interact between cell bodies, producing collective movements. So we will build our model of propulsion around this mode of movement, the COPG. We'll propose a psychophysics model. We'll look at these oscillatory movements as a small subset of behavior, but allow us to view the COPG circuit. And the psychophysics models that we'll propose will account, hopefully, for a wide range of behaviors outside of these oscillatory movements. So this is an example of a CPG. So the COPG is based on a CPG, or a collective pattern generator. And so this is a circuit of a collective pattern generator in a neuronal system. So we have our neurons and we have our synapses here. So we have this network of neurons, and they're both inhibitory and excitatory synapses that play a role in setting up this collective pattern generator. And the collective pattern generator generates rhythmic motions such as insect movements of their limbs, also other types of pacemakers in the body. And so we can use that model. This is, of course, a neuronal model. But if we think about this now more broadly into the COPG, which is a collective pattern generator, we can think of this in terms of different the cells being connected by sort of these points, these mucilage trails, and then they have a relationship between these different points that we can either arbitrarily measure or measure on the cell. And so we have these weights that vary by how the thing is moving, how the cells are moving against one another's strengths. There's like a weight of a certain strength between the 2 points. And so that gives us some way to model the movement of the colony. And so we can use a series of coupled sinusoids to model the ideal collective pattern generator. We can model like a pure sinusoid and then have overlapping sinusoids representing each cell, and then vary the distance between these overlapping sinusoids so we can get different degrees out of phase behavior. This simulates coordination or discoordination. We can also add noise to these sinusoids so we can get noisy sinusoids, which is helpful in some of the modeling the different phases of movement and maybe some of the noise that we observe. We can also look at the speed of movement, or the derivatives of cell position. And so in the paper I put in the chat, we actually did some measurements of this movement in terms of its speed and its velocity and all of that. And so this can vary according to the effects of any given stimulus. And so one of the examples here are differences in stretch phase. So here's a colony and it's moving. You have this leading cell, this lagging cell, and then these intermediate cells. And you can see that when it stretches out, it doesn't just stretch out back and forth. These cells kind of bunch up about mid-movement. And so this can be modeled using this type of computational representation. We can also look at sensory thresholds. So in this case, each cell is receiving sensory inputs, which are these eyes, and differences in light intensity are measured by output movements in the weights. And so we can give it a certain amount of light input, and it affects the movement of this network. We can also look at the signal to noise ratio. So this is useful in the dynamic sensing of water column physical properties, and that can also be measured by these output weights. So thank you for your attention. I just gave, I think, a short version of this idea. I'd be interested to see what you think. And if you like what you see, we can collaborate. Our group, DevaWorm, is a very highly collaborative group. We're a virtual group. We have a website and a series of GitHub repos. You can check those out. We have our weekly meetings on YouTube, so you can check those out. And I'd like to thank Thomas Harbeck for his observations of diatoms. He was the source of a lot of the videos shown in this talk. And check out his website as well. Thank you.
Thank you, Bradley. That was a really interesting and out-of-the-box talk. So that was fantastic.
Thank you.
So I have a couple questions from Najee. She's back online, but she's on audio with us. Najee, do you want to ask the question by yourself? Unmute and ask.
Sorry, she doesn't have a microphone either.
Okay, so I'll read the question. She asked, maybe because she missed the beginning a little bit, but she was asking, do you know the mechanism involved in to the sensitivity of the carbon dioxide? Are they sensitive to pH? Do they express channel involved in CO2 or O2 changes like the task channel?
I'm not really familiar with the biochemistry of it, but I mean, I know that they do have, I think it's probably somewhat similar to that, although I don't really know that much about it.
So I have a few questions, but I think I'll start perhaps with the title part, probably the more controversial part, right?
Yeah.
So which is are you equating cognition to just movement here, right?
Well, I mean, I guess the idea of cognition is information processing.
Mm-hmm.
So the idea is that you have movement, which is an output of what it's gaining. So it's getting sensory inputs, and there's some process within the cell, and it's producing an output.
Right.
And so the movements but the movements, of course, can also be sort of, rhythmic, just kind of a set pattern, and then the inputs are changing that. So there's some information processing going on there. We don't know exactly what it doesn't necessarily matter what the mechanism is. It's just kind of a way to think about it. And so, that's the kind of thing, like it is kind of a metaphoric approach.
Right. Yeah.
But you can also use that paradigm to kind of build models around it and see the movement in a new light.
Right. I mean, I would say the same. We look at animal behavior, for instance, but we are using movement as a way to figure out if they're able to do something or not, right? So it is a element of cognition. That's why I was curious about your movement statement, because that's what mainly you were showing in your phenotypes there, which is really cool. I have one other question, but that's more related to the extracellular matrix. So maybe I'll come back to that in a bit. We have a question in the chat. What is the limit of non-neuronal cognition?
Well, the limit, you mean in terms of like the complexity of the behavior, or?
I assume so. Maybe we'll get a clarification in the chat, but I think that's a good start.
Yeah, I think it's probably, it's a metaphor. You want to make sure your metaphor is somewhat rigorous. So you don't want to just call anything like cognition. There was a recent paper by, it was called Cognition All the Way Down, where they argued that cognition exists in cells and different subcellular mechanisms. I think, like, if you observe in a cell where you have these sort of intentional movements, I think you can interpret it as cognition. I'd be very careful about doing so. But I think the limit is basically where you have an input some process internal to this thing, this cell or this organism, and then an output that's somewhat intentional. So, you want to distinguish between sort of stochastic movement, maybe like in maybe like Brownian motion, or something more intentional. And then that's where you kind of get into the definitions and kind of quantifying it.
Right.
So that's the challenge too.
Yeah. So maybe I'll piggyback on that with the extracellular matrix, because you're talking about how that's allowing them to tag with each other. And presumably that's dependent on the input and the output, right? So they're basing it on how they're interacting with each other, sensing their environment and putting those things down. Do we know much more about the intentionality there in terms of laying down that extracellular matrix?
I don't think it's very clear what the intention. I mean, they definitely generate this stuff, and then it's sort of sticks together. So there's probably a lot we don't know about, like what exactly is going on there. They do actually move, of course, intentionally, and the colonies actually fall apart after a while. So, we don't really know what's going on there. There could be just environmental factors at play where, the environment gets to a certain point where they just, or their age wise, they kind of fall apart. But there's definitely like a lot to learn there. There isn't a lot known about like some of the things I mentioned here, which are kind of speculative, but I think it's a really interesting system.
Right. Yeah. Perhaps one last question before we have to wrap up. This is also for the question answer session. Can I also ask about the upper limit? As in, what's the most complex, sophisticated behavior an organism can do without neurons?
Well, I think it depends on like maybe, collective behavior is interesting because, like with bird flocks, for example, you have these and they have neurons, the birds have neurons, but the flocks don't have neurons. And so they have to coordinate their behavior in a way that goes beyond sort of the neuronal, standard neuronal suite. There's a lot of I mentioned stigmergy in the talk, and that's an idea from complexity science where they like a bunch of organisms, say like ants or something, will set up a bunch of things in the environment. So like these extracellular matrix trails, and they use them to read the movements of other organisms. And so ants can build trails through pheromones, laying them down and then having their neurons interpret those as like the way to go. And so you can do it that way too. And that makes for a lot more complex behavior than you would get from just a single brain.
Right. Yeah. Fascinating material. Both of you, I really enjoyed this session today. I'm glad in a little way that we were able to talk a little bit more because we missed out on our second speaker today. But this is fascinating stuff. We will post some of the links are on the chat, so feel free to follow up and perhaps communicate with Najee and Bradley through online mechanisms, because I think that's one of the cool things about being online, right? So they're able to do these and continue these discussions. So thank you again. This was absolutely wonderful and fun, and enjoy the rest of NMC. I'll see you soon. Thank you, William, for hosting us.
Authorship position of henceforth women-led papers has steadily increased over the last 2 decades in 5 broad scope neuroscience journals. This categorical authorship gender breakdown can serve as a proxy for the field of neuroscientists defining categorical citation base rates if gender is not a factor in citation choices. A recent study by Jordan Dworkin, Daniel Bassett, and colleagues investigated whether citations in the 5 broad scope neuroscience journals reflected these base rates. Instead, articles published by men as the first and last authors, henceforth articles, have been cited more frequently than the base rate, whereas women-led publications have been cited less frequently than the base rates. But this, of course, is a broad view of a diverse field with many subfields. And so we asked whether this trend generalizes to more focused domains where base rates and citation practices may look different from the field as a whole. We focused on the subfield of cognitive neuroscience, and we reviewed authorship and citation patterns in the subfield's flagship journal, the Journal of Cognitive Neuroscience. In the last decade or so, as we can see here, the categorical gender breakdown in authorship has generally been stable with a slight upward trend in the proportion of WW papers. Also noteworthy is the fact that the percentage of women-led publications has been markedly larger than that of the broad scope journals. As with the broad scope journal investigation, we'll define citation base rates according to this categorical authorship gender breakdown in JOCN. But before we can reveal the journal citation practices, I'll step back just a bit and explain the approach we took with the help of a talented undergraduate, Larry Akinola. I'll briefly describe the method, saving discussion of the more nuanced details for the discussion period. So we downloaded and organized metadata for the 2,106 research articles published in JOCN from January 2009 to July 2020, a snippet of which can be seen here. These metadata were combined with the metadata from the 5 broad scope journals analyzed previously. We then probabilistically determined the gender category of each JOCN article. In an example article, the first author was named Simon and the last author was named Sabrina. These names were queried within the Social Security Administration's baby name dataset, and if they weren't found there, through genderapi.com, which relies on census and social media data. And that allowed us to obtain the probability of the name belonging to a man and woman. If the probability of belonging to either gender was 0.7 or greater, that gender was assigned, and so this article was assigned as a man, woman, or MW article. We did this for all JOCN articles, and that yielded a new column of authorship gender categories, which served as our expected base rates for citation, and that gave rise to this plot that we saw a moment ago. We next determined the gender categories of the papers cited by JOCN articles. So JOCN citation lists, as seen in our example article, include only first name initials. However, the metadata included DOIs for each article's citations. Similarly, each of the articles categorized in the previous step had a corresponding DOI. So therefore, we used the column of DOIs as a lookup table for each of the citations in the reference lists of the JOCN articles. When a DOI was matched, we categorized that citation according to the gender category assigned in the previous step. Inspection of the citation rates reveals that women-led papers have been cited less frequently than the base rates and vice versa for MM papers. But it's important that we put these citation patterns into context by explicitly factoring in the base rates in order to determine the true extent to which citation patterns are imbalanced. Toward that end, we'll label the citation rates as the observed proportions and the base rates as the expected proportions. For each category, we then compute a gender citation balance index by subtracting the expected proportion from the observed proportion and divide by the expected proportion. A value of 0 indicates a citation rate as predicted by the prior, with positive and negative values indicating over and under citation compared to expected, respectively. So for JOCN, MM papers had a larger observed proportion than expected, yielding a positive index. The remaining categories all had negative indices, revealing an undercitation of women-led papers, with the WW category being the most undercited. Finally, we can break out by author gender category, and we see that the indices for citation lists of MM authored papers show a trend much like the overall trend we just observed. However, this group is not solely to blame, as we see a pattern similar for WW authored papers, as well as for the mixed gendered authored papers, albeit with minor variations in magnitude. So we have shown that gender imbalances in citation practices of neuroscience generalize to the subfield of cognitive neuroscience, and the pattern of these imbalances is consistent across authorship gender category, implicating systemic factors. So what's a field to do? Well, calling attention to findings like this is a great start. And finally, I've spent the past month or so developing a web tool, which is shown here, in which authors may submit a reference list and receive the gender category breakdown and gender citation balance indices in return. Here I submitted a reference list from a paper that we published earlier this year. And as you can see, it's coming. I too am guilty of citation imbalances in the same direction. So Dr. Brad Postal, in whose lab I work, is assuming the editor-in-chief role at JOCN and will be encouraging authors to use this tool to see how their reference lists stack up as a first actionable step toward improving the citation balance. And I now look forward to our interactive discussion, and thank you very much for listening.
Owing to the online nature of this conference, this is the applause you hear from the attendees. If you had excellent work, Jacqueline. So we are waiting for questions. As we are waiting for questions, I'm just trying to see if I can quickly post this link to our attendees in the chat. Just give me one.
Sure.
Yep, perfect. This is great. And thank you for actually providing this tool, because this is what has happened. Like, as I'm listening to you, I have this question like, okay, well, I need to check how my citation practices are. And I've seen that there are some tools that are being developed, but they were like in test case, or like it's still not ready for the real world. But this is great. This is really good.
Well, I will say that this just has become, live, and so we're still in the process of working through it. But it does seem to be working well. And what we've found is people who have tested it, they've had the same sentiment. I'm curious to see where I'm at, and it seems that the majority of people so far have more or less this pattern. So,
Yeah, I mean, I can. Yeah, I can imagine, given the strong trend that you show across all these participants.
Right.
So I think we have a question.
Okay.
Oh, yes. So we have a couple of questions. First one is from Don Taylor. Great work. How much do you think the men-women imbalance of speaker selection at conferences impact awareness of men-women research, leading to this bias?
So that's an interesting point. So we're here really just to kind of shed light on these patterns. So we are not social psychologists, so we can't speak to all the potential sources of this, but certainly that could be one issue contributing. So when work is disseminated at a conference, it may get a little more feedback and press, and then make it easier to publish. So that could be one of many factors, certainly. And so the goal would also be to increase representation at conferences as well, and make sure that there's more dissemination of the results across all the different gender groups. Yeah.
Right. Yeah. We have the next question, like, very interesting. Thank you. I wonder if some of the bias may stem from the traditional overrepresentation of male scientists. That is, do you give citations of old and new papers the same weight, assuming old papers were more likely to be authored by men?
Yes. So we purposely, for this analysis of JOCN, focused on the articles published within the 2009 to 2020 time range. Now, this, of course, means that there still could be references to much older papers, but as we've seen, at least in JOCN, the authorship is generally stable. So it's kind of in a point where the field is not changing all that much. And if people are tending to cite more recent papers, then, they should be citing kind of more at this level. What seems to just be happening is that, people in JOCN are citing the broad scope neuroscience journals in general more frequently than JOCN. And as I mentioned in passing at the beginning, the gender balance in the 5 broad scope journals is more skewed towards males. So I do think, yes, citing older papers would tilt the balance more in that direction, but also simply just citing outside into the broad scope journals is going to tilt the balance as well. So and that's why we're pushing towards looking at these indices, so balancing by the prior. So if your group does tend to be more male, that should be taken into account as well, that we would expect you to also cite more male papers.
So we have a question from a panelist, and I would recommend Jorge, you can unmute yourself and ask the question. So Jorge, are you talking? We are not able to hear you.
Oh, sorry. Can you hear me now?
Yes.
Yeah. Great work, Jacqueline. I was wondering what percentage of authors' genders were not able you were not able to classify, because you mentioned the 70% threshold for classifying them.
Yes. So in the JOCN analysis, we, through the first pass, had less than 10% that, for a variety of reasons, weren't categorized. In some cases, we had single author papers, and so we then categorize them as either male, or W, women papers. But then we did have a few cases where they weren't categorized. So in that case, because it was so few number of papers, we were able to visit faculty pages and journal websites. And if there were pronouns available, we were able to categorize those. So for the JOCN, it was maybe only a few authors in the end. For a much larger data set, for example, the 5 broad scope journals, then you start having many more articles to work with. And so in that case, I don't recall off the top of my head, but I think it was still about 10% or less that weren't categorized because of the probabilities. So
that's great. Thanks.
Mm-hmm.
We have one more question from Eleanor. Could you test if scientists also have a biased view of other scientists in the field, maybe with a questionnaire, meaning that they know more male than female scientists than the real ratio? This could explain why they cite males more.
So the previous study that I mentioned by Jordan Dworkin and Daniel Bassett and colleagues, they looked into what they called social networks. So they actually factored into these kinds of analyses for the 5 broad scope journals, the collaborators and people in their academic networks. And they found that even when you account for who people know and who people work with, this pattern of imbalances still emerged. So it's something beyond just who you know and who you happen to work with.
Great. I'm just sending this information to the attendees. Great. So Dan, why don't you unmute yourself and ask the question?
I guess I can. Yeah, it's really interesting. I wanted just a quick question about sort of the practical moving forward, knowing these imbalances exist. Like, what do you propose we do at the journal level? Should reviewers flag this if they notice there's a gender imbalance, or what do you recommend?
So if Brad Postal's here, he may have a little more to speak about that. But what he is hoping to implement at the Journal of Cognitive Neuroscience is to encourage authors to use this tool. It's not going to be a mandatory requirement. And then they would report the outcomes from the tool, these boxes that we see here on the slide. And the idea would be that reviewers would see this information, but it would not be a factor in whether or not to accept or reject the paper, but rather if there is a strong imbalance, perhaps recommending some additional references that improve the balance, for example. But there's no penalty. It's really just a way of making people aware of this and potentially seeking out more of the literature that they can include that's relevant for their topic. And the hope is after a year, we could do an analysis to see whether this kind of tool is having an effect. So
it looks like Brad has his hand raised, so I can allow him to talk. That's okay?
Mm-hmm.
Yeah, sure. But it'll have to be pretty brief, unfortunately.
Mm-hmm.
Okay. I think you're, yeah.
Yeah, Jackie actually said everything I was going to say. Thank you, Jackie. Wonderful talk.
Thank you.
All right. This is a very interesting and important area of concern for us. So we have a lot of questions, but unfortunately we have to move on to our next speaker. Jacqueline, in the meantime, can you try to answer them using the Q&A? That would be really helpful.
Sure.
So we have our next speaker, Garrett Flynn from University of Southern California, who will be talking about stakeholder perspectives on cognitive effects of brain stimulation. The floor is yours, Garrett.
All right, yeah, thank you. So as some of you might know, brain stimulation devices are increasingly used to treat neurological and psychiatric conditions. We have things like deep brain stimulation for Parkinson's disease, transcranial direct current stimulation for depression, motor brain computer interfaces to restore the function of lost limbs. And the lab that I work at USC is developing a memory prosthesis for the treatment of dementia to restore memory encoding. And regardless of whether the intended effect of the device is to modify cognition, there tends to be unintended changes to language, memory, and other cognitive processes that is understudied and underreported by both patients, caregivers, and healthcare providers. So the goal of this literature review was to characterize the cognitive effects of brain stimulation in previous literature. So we conducted a PubMed database search for qualitative studies on brain stimulation. This ended up returning about 10,000 articles. We screened most of these in the titles and abstracts, had about 76 full texts, included about 41, which comprised 36 total data sets. And we found about 15 papers from 14 data sets that did include mention of cognitive effects. So this was sourced by sort of a primary search on brain stimulation with the conditions of perspectives, opinions, views, interviews, and qualitative. And then we just conducted a secondary search to make sure that we caught all these specific devices. So that would include responsive neurostimulation, transcranial direct current stimulation, all of the others I had mentioned previously. So some examples of the returned articles included patients with obsessive compulsive disorder who had mentioned, for instance, yes, I can pay attention somewhat better when I have the deep brain stimulation device, somewhat better at focusing at the task at hand or at music while playing in a band, because normally distracted with my thoughts, and then I could not follow the story anymore. And so as an example of the way we code this article, this is an actual effect experienced by somebody who has a deep brain stimulation device. To some extent, cognition is restored to sort of a baseline level before the disorder. And it's a general cognitive question. So it's in response to something that the researchers had asked. It's not sort of spontaneously reported by this patient. On the other hand, we do also have examples of healthcare providers who were asked about a speculative neurotechnology. So when asked whether it would be ethical to provide surgical and memory enhancement, if a patient should request it, 48.6% of respondents said it would not be ethical. And so this is an expected effect from healthcare providers who are asked about enhancing cognition. And this was specifically about a cognitive enhancement question directed at these healthcare providers. And so going through the 14 data sets that we did have, we found that patient-reported cognitive effects or expectations were present in about 7 of 14 data sets. And only one of these 7 data sets was sourced by asking patient about such effects. So this means that most of the patient-reported cognitive effects were just spontaneously given by a patient. It's not something that the researchers were specifically looking for. And these responses, which generally were from patients with Parkinson's disease, were largely negative. So they were cognitive effects that were detrimental to the patient's well-being. On the other hand, provided reported cognitive effects or expectations were present in about 5 of the 14 data sets, and 4 of these data sets asked providers directly about cognitive enhancement. So there seems to be a pretty big focus on sort of speculative devices or devices that could restore function beyond some degree of baseline. And this is sort of incongruous with the actual patient-reported cognitive effects that exist. So what this generally shows is a current reliance on healthcare provider perspectives, which leads to underreported negative cognitive effects for patients, and lack of understanding about the patient perspective on early stage devices and the possibility of cognitive enhancement from these. So we don't know what patients would even want from such devices if they have any concerns, and whether we might focus our resources on more conventional like symptom management rather than sort of these more speculative devices. And to address these problems, we suggest that future neurostimulation studies should more consistently and rigorously evaluate such patient perspectives on potential cognitive effects. So in the neuromodeling and interface lab, which is building that memory prosthesis, we're currently in the process of conducting qualitative interviews with trial participants, as well as individuals with mild cognitive impairment and individuals with biomarkers for Alzheimer's disease in order to develop a qualitative tool that allows us to gain a grasp on these intended cognitive effects, which could generally be used in this sort of growing field of cognitive neurostimulation. And so, yeah, I'd like to thank the people in the lab that really helped with this. Dong Song is the principal investigator, and I couldn't have done this without the support of some great high school students, Kevin Rao and Miranda Zhang, and external collaborators from the University of Washington, particularly Aaron Klein. So yeah, thank you for listening, and that's
all right. Thanks for the wonderful talk, Garrett. So as we are waiting for questions, I have a more like I would say, like, how would we even start asking this question to like the patients? So for example, like what would be like, is there any, do you think it would be like questionnaires, or should it be like tasks that we actually assess if there are changes? So what would you like suggest? Like, is there a questionnaire that we could use?
Yeah, so for at least the literature review, we focused on qualitative responses. So that would not include things like generalized memory tasks or other cognitive tasks in the clinic. We sort of steered away from those. Surveys are also notoriously difficult to get any sort of deeper response, and it's generally sort of leading questions that may or may not allow for really in-depth, open-ended responses. So I would say that the suggestion there would be semi-structured qualitative interviewing. Yeah, so getting the direct responses of patients, asking about any sort of changes in their cognitive processes, more directed at sort of specifics like language and memory.
Got it. And I think you briefly touched it before, but I didn't quite grasp it. Like, was there any number that you could tell that the patients were expecting cognitive improvements, like an expected outcome of the treatment, or did you find any?
Yeah, so that's slightly showed on this graph, or the top figure. So there's one summary of an enhanced effect or expectation from patients themselves.
Okay. So this was expectation, not necessarily the outcome that was.
Yeah, I would say that we didn't find any enhancive effects that were actually like reported as testimonials from patients. Yeah.
Okay. And so just wanted to let the attendees know you can always ask questions and we can bring you on screen. As we are waiting, I have one more question. So was there any, like, I'm curious if how much of this is actually from, say, deep brain stimulation and how much is from like non-invasive brain stimulation?
Yeah, so there was a pretty heavy weighting towards DBS. So the 3 devices that we actually did gather in this literature review, or that were the results of the sort of screening process, were deep brain stimulation, transcranial direct current stimulation, and one bidirectional brain-computer interface study. The bidirectional study was actually not included in the cognitive effects, so there was no cognitive effects reported there or asked about. And I think transcranial direct current stimulation was about 1/3 of the studies, and DBS composed the other 2/3.
Okay, nice. Yeah, Jorge, please unmute yourself and ask a question.
Great, thank you. Great work, Garrett. I'm curious about the caregivers and the family bars in this plot. It seems like for some of these things, like placebo effects or like heightened expectations of getting better, can get in the way of patients and providers. But it seems to me that caregivers and family could be a better witness of like actual improvement. And I'm wondering if this is something that can be enhanced, or if for example, I wonder why there are like expectations bars for caregivers and family. Like, do you think they are not asked before the treatment what they're expecting? Or because I think that will be like valuable information.
Yeah, 100%. I think that the patients and caregivers, or the patients' families and their caregivers, are very important repositories of knowledge about what actually happens in daily life. I mean, if brain stimulation modifies cognition, then to some extent, the reports of patients are slightly suspect. They may be tinged by the stimulation itself. And so caregivers and family members can report on changes in behavior that the patient may not themselves be aware of. Yeah, what I would say is just, yeah, I think it's important that it be considered in the future. I'm not 100% sure how it can itself be enhanced, but I think it's, yeah, certainly worthy of note. And yeah, in this, it's largely reports of worsened cognition as well. So I explicitly remember a spouse of a patient mentioning that her husband felt like one of her pupils at school. Like, she's a school teacher, and he felt like he was just like one of the distracted students all the time. And so that was interesting. But I think to some extent, it's a little fuzzy still. And I think there needs to be a lot more work done there.
This is getting a little detailed, but were there any instances where you found like conflicting reports? So if, for example, you see that public, there were 2 reports, and were they the same from someone who had a family or a caregiver give a rating?
Sorry, can you repeat that question a little bit? Are we comparing public and caregivers? Is that?
Yeah, so are they from the same, is it about the same study? Is it from the same study that you...
Oh, okay. Yeah. So if there was a mixed study, they were largely healthcare providers and caregivers and family members or patients. The public itself was only queried in one of the research articles. And so there really wasn't a mixing between those like public and researchers were 2 sort of independent groups that were characterized, and then the others were sort of connected together in some cases.
Okay, got it. Yep. Okay. If we have no more questions, let's thank Garrett again, and we'll bring on our next speaker, Jorge Morales from Johns Hopkins University, who's going to be talking about challenges for decoded neural feedback.
Yeah, thank you so much for being here from wherever you are. This is a purely theoretical work that I'm presenting for the first time, so I'm really eager to hear some of your feedback. When neuroimaging allows us to make correlational claims, we can correlate what's in a subject's mind with what's in their brain activity. Causal explanations, however, are necessary for truly understanding neural mechanisms, and interventions are the hallmark for establishing causal relations. However, neural interventions, especially in humans, are complicated because they can be quite invasive, non-specific, and sometimes even both. Decoded neurofeedback, or DECNEF for short, it's a new protocol using fMRI and BPA that allows us to decode brain activity and with subject self-regulation alter patterns of bold activity in a directed way. So DECNEF allows us to make causal interventions that are non-invasive, functionally specific, and even unconscious. So it's an extremely promising new technology that neuroimagers have available. Let me just go quickly through the protocol of DECNEF. It consists on 4 phases. First, there is a pretest where a baseline is established for some behavioral outcome. For example, the mean skin conductance response for seeing fear-inducing stimuli. And then a decoder is constructed. Subjects are put in the scanner and their brain activity is measured, and a pattern is, a decoder is built for classifying what arbitrarily is labeled as a target or a non-target among the stimuli. And then the crucial phase comes up, and it's the induction phase where subjects are brought back to the scanner and asked just to try to enlarge an indicator on the screen, like a green circle, with their minds. They're not given more instructions. They're just told, try to make this circle bigger. And as they're trying to do that, their neural activity is being measured in real time, and a decoder, it's quickly decoded. And that decoding, that pattern of activity is compared to the pattern of activity of the target stimulus. And depending on how similar these two patterns are, the one that they're experiencing at the same time in real time with the target stimuli, the feedback is provided back to the subjects in a proportional way. So how much they can enlarge with their minds the circle, it's really a measure of how similar the patterns of activity that they're currently experiencing are to the target patterns of activity. And they do this for several days until they achieve a certain level of similarity. And finally, if this intervention worked, in virtue of being exposed to these unconscious mental states pertaining to, let's say, the target snake, in the case of a skin conductance response study, if the intervention worked, they could see that the behavioral measure changed for the target stimulus, but not for the non-targeted stimulus. So this is extremely promising technology. There are multiple applications for cognitive and clinical domains, and there are, in some cases, very long-lasting effects. They can, the changes sculpted into subjects' brains can last for even 2 months at least. There are 3 challenges I just want to bring up for discussion that this technology, however promising it is, raises for neuroimagers, philosophers, and ethicists. Challenge number 1 is a general VMPA worry, and that can be captured by what these authors call the decoder's dictum. According to this dictum, if information can be decoded from patterns of neural activity, then this provides strong evidence about what information those patterns represent. But this dictum, it's false. We really can't infer from our ability to decode something from the brain what exact information is actually being used by the brain. Second challenge is that there is no one-to-one mapping between voxels and neural activity. Even if you target a particular voxel pattern, that doesn't mean that, and you match it during induction, that doesn't mean that the underlying neural activity, it's exactly the same. You could have the same voxels being activated with very different neurons, maybe even different neural populations being responsible for getting those voxels. And therefore, when you try to read out, but also when you try to write back into the brain a particular pattern, you could be doing something extremely different that has nothing to do with the target. And finally, I think that we risk actually creating unintentional interventions that, that the experimenters are not aware of. When everything goes right, you read off snakes and you write back snakes into the brain, but because of the previous 2 challenges, we really don't know exactly how these mappings work, and we don't know exactly what information we're reading and writing back. And therefore, if we focus just on writing back information, we could, instead of writing off a snake, we could maybe activate a pattern of activity of something similar, long, kind of stringy thing that it's, instead of a snake, a murder weapon. Or something else that we actually don't know precisely because we don't know exactly what information is being written off. And these could have like cascade effects or downstream effects, because even though these interventions are really targeted for one region, we don't know what kind of effects can happen if you target, let's say, visual cortex. There could be memory effects or cognitive control effects that it's hard to predict. That are hard to predict. So just in conclusion, I think that TechNev is an extremely promising technology. I think it's one of the most exciting things that has happened in neuroimaging lately. It allows us to, it has cognitive and clinical applications, and it allows us to do causal interventions that are specific and non-invasive. But these 2 great properties of TechNev are also, can also be called into question because some of its mechanisms remain obscured, and at least potentially they could even be risky. So, especially because there could be long-lasting effects. So, thank you so much for your attention, and I'll be happy to talk about this more.
Thanks for the wonderful talk, Jorge. As we are waiting for questions, I have a question. So, does this approach approximate what people use as like exposure therapy for, let's say, treating PTSD? the patients are actually try to relive the experience, right? Is
Yeah,
that
this is
similar to
great. This is, yeah, no, absolutely great question. One great thing about DECNEF is that literally what you just say, it's something we can do, right? Like one way of treating phobia, it's by re-exposing patient over and over again to the feared stimulus. Or if someone has PTSD, you can be over and over exposed to the traumatic experience. A huge problem with that is that the treatment itself is traumatic, right? Because you have to be re-exposed to what you fear, what you're traumatized by, and this even though it's a very effective treatment, it has problems with there's a lot of dropout and it's very stressful. So Vincent Taschereau de Michel and colleagues recently published this paper that it's really interesting because they were able to use surrogates to apply their decoders to. In this case, they weren't patients, but like subjects that showed more phobia to certain stimuli. But you could think about using these in patient population as well. And the idea is that you show this fearful stimuli to surrogates, but not to the patient. And you build a decoder with the surrogates information, and then by using fancy technologies like hyperalignments and such, you can use that decoder to in the induction section with a patient. So basically, you can expose patients unconsciously to their feared stimuli, and actually, at least in these laboratory conditions, there was a reduction in the skin conductance reactions for patients who had never seen the snake before, with a feared stimuli, whatever it was, without actually being exposed to it. So yeah, it's very promising. As I said, it's an extremely promising technology.
Oh, that's really interesting. Yeah, so that's a very interesting way to approach this. But like you mentioned, the first thing, the MVPA, it's still kind of a black box, right? So we do not know what's happening and what is changing.
Yeah.
So do you think if we have, let's say, instead of MEPA, some other, like a machine learning model where you understand what is going on inside, would that be a better option in this case?
I would say, I mean, of course, the more we understand about a decoder or any kind of machine learning algorithm, of course, it will be, it will tell us more about the information that it's being read off or written back into the brain. That said, I think that there's a huge gap in our understanding of what neural representations are. I think that we talk about neural representations as something we understand, but it's kind of up in the air, like what really a neural representation is. For example, we know that there are some correlations, like you show a subject like a Gabor patch oriented in a particular way, and then you see some neural activity that seems to correspond to that. But information in the brain is like widespread, right? Like probably with a sufficiently sophisticated algorithm, you might be able to decode almost any kind of information from almost any kind of region in the brain. And that's very unlikely. It's very unlikely that that's information that the brain is actively using for representing that stimulus throughout the, let's say, neural cortex or something like that. So I think there's a long road to walk in order to understand exactly how the brain represents things. And almost like what makes DECNEF exciting and promising, it's also something that should give us a little bit of pause in terms of like giving these super hardcore reinforcement to subjects, some of which have up to 2 months have been tested, and the reinforcement like still have its effect. Because I mean, I don't want to sound like scaremongering or anything. This is more like a theoretical question, but it's not too far-fetched, I don't think. Like if we really don't know what we're doing in terms of altering neural representations, we might actually do something that has some unintended consequences.
Good. So I have one more question.
Could I ask
has,
one more question?
yeah,
Sorry.
go ahead. Yeah.
Yeah. So which regions are you specifically aiming to target with this technique? Do you like aim to focus on, say, subcortical arousal areas to deal with phobias, or more object recognition?
Yeah,
ventral
that's
vagal
great.
cortex?
So the way it has been done, it's been a few areas. The decoding from basically anywhere else from visual cortex or the motor cortex, it's actually pretty hard. Decoding from PFC, for example, yields like really low, like barely above chance classification often. So at least, for example, the study that it's on the screen right now, they actually targeted visual cortex. They decoded patterns in visual cortex to kind of like expose visually without subject's awareness to the fear stimuli. I don't know about subcortical. I don't know. Yeah, nothing about decoding on the subcortical areas comes to mind right now with fMRI. I think so far it's mostly being done with cortical structures. Some of them in PFC for sure, and maybe parietal cortex, and definitely visual cortex. So it's early times in this technology. Yeah.
Garrett has a question. Garrett, why don't you go ahead?
Yeah, Jorge, I just want to say this is a great talk, and I think this technique is super interesting. I just wanted to ask, like, how long does it take for patients to train up on this control task? And how long does it take either like during that or after that to see clinical effects? Yeah, so it's usually like about a week, like maybe an hour session, hour-long sessions, about 5 days, where you do this induction task, where you just try to enlarge this circle with your mind. That's about what it takes to then see some measurable effects in whatever measure you use. I mentioned skin conductance response, but it's when you also, for example, accuracy for Gabor patches, like literally how accurate you are at discriminating on left or right tilted Gabor patch was the first Deknev paper. One of the orientations of the Gabor was like reinforced for a week in subjects' brains. So it was as if they were more familiarized with that orientation. And then when they did the task, they improved their accuracy in discriminating that orientation, but not the others. So in terms of actual clinical applications, there's been only a few with functional connectivity Deknev for depression patients, for example, where after using a battery of tests, they showed that depression symptoms were reduced. And that's the one that actually showed like up to 2 months of an effect. Like the improvement lasted for, in depression symptoms, lasted for up to 2 months. So that's about it. Yeah.
All right. If we don't have any more questions, okay, so there's one question from Eden. Besides snake and animals, are there other fear that you consider to cure?
Well, I don't know if we can actually cure phobias with this technology yet, like in actual patients. This is still somewhat speculative, even though it has been shown to work with lab subjects. And I think that the idea is like anything that requires like extra exposure or extra familiarity with a particular type of stimulus should in principle be a good target for Deknev. Anything that requires you to be familiarized or overexposed to. So yeah, any kind of
Got it.
Yeah.
Yeah, I think we are at the end of our session. Let's thank our speakers again, and thank the attendees for joining us. And yeah, I think that's about it. Thanks again.
This was great. Thank you.
Yeah, thank you. Thank you for hosting. It's been a great time.
Hey everyone. So in today's session, we have 3 speakers. We're having a brief technical issue with our first speaker joining us. but let's just give it another minute to see if we can have him join. So I'm just trying to reach the speaker. If this doesn't work in a couple of minutes, we'll just have another speaker present instead. So let's see. Oh, so Harman is in the chat. Can we, Elnaz, who's our backend host, Elnaz, can you help invite him on screen so that he can present? So Harman has been invited. Harman, if you unmute yourself and share your video, you should be able to present now.
Hello, everyone. Sorry about that. We had a small mishap with the Zoom accounts.
Well, it's great to have you here. Let me just quickly introduce you. So our first speaker is going to be Herman Espinoza. He is going to talk about leveraging spatial connectivity for more efficient planning in naturalistic scenarios. He's currently a CS PhD candidate at Northwestern University working with Malcolm McIver. And without further ado, I'll just let him present.
Hello, everyone. Thanks for being here. Sorry about the little mishap, and thank you for the introduction. All right, so I'm Herman, and I work with Malcolm McIver at the Center for Robotics and Biosystems at Northwestern University. Our main research focus is in planning, and I want to start with a big disclaimer because I know we have a few neuroscientists here. We don't know how planning is done in nature, and we don't know if biology solved this in a complete different way, but we have a very strong well supported hypothesis that claims that the brain uses the information available and its knowledge about the environment to create a prediction, to create a tree of predictions, and then uses those predictions to select actions that will eventually maximize the probabilities of achieving a goal. We will assume this is true for this talk, even though we know it's controversial still. We will assume it's true for this talk. Then the next question is how big this tree of prediction has to be able to effectively plan, and what it should cover. And in this talk, what I will show you is how the volatility of the environment and the uncertainty about the present increases the number of predictions necessary to effective planning. And I will also introduce you an approach that is theoretically possible in biology to drastically reduce the number of predictions while still maintaining similar outcomes. One of the aspects that increases the complexity of planning is the volatility of the environment. In a static environment, it's easy to create the right predictions, right? Because nothing changes. And those predictions can help us create a perfect plan. And then we can just follow it to conclusions because the environment will be the same. But if in a changing environment, making predictions is a lot harder, and we must explore all possible values of every single possible variable that we cannot predict. And this grows exponentially with the numbers of actions into the future. So that means if I don't know if something is going to happen or not, I need to plan for both the scenarios. In other words, the less certainly we can predict the future, the bigger our prediction tree is going to be. But what happens if we're not even certain about the present, right? Oh, let me explain what I mean with this. Let's say in a naturalistic environment, during a predator-prey interaction, the prey goes behind an occlusion and temporarily loses sight of the predator. Using just the current information, the prey could assume the predator is no longer present, or it stays static on the last known location. We know both assumptions are really detrimental to its chances of survival. But something better the prey could do is use its knowledge about the environment and the predator and guess possible current locations with some level of certainty. We call these guesses a belief state. And in this case, we can see it as a list of possible locations of the predator and the associated probabilities. Once built, the belief state can be used to create multiple trees of predictions and then combine them in one big tree to determine the best possible plan. However, this means the more sparse the belief state is in the space, the more predictions the prey will have to explore. In other words, the less certain we are about the present, the bigger our prediction is going to be. As we've seen so far, our knowledge about the present and our ability to accurately determine the future impact the number of predictions necessary to plan. And I'm going to give you a practical example with a simulated experiment we did, just to show you how this works. In a square grid, we located a prey and a predator agent. And sorry, 1 second. There you go. We provided the prey with planning abilities, and we reward it only when it makes it to the goal in 50 steps or less. The predator has two behaviors, search and chase, and it's averaged 50% faster than the prey. Something important is that both behaviors are stochastic, so the prey cannot predict those deterministically. And finally, we added occlusions that limit the movement of the agents and also their visual fields. So they can only see each other and know where the other agent is when there's an open straight line between them. This introduces the concept of partial observability, and it forces the prey to create a belief state. For this very simple scenario, right? If we want the prey to visit every single possible prediction in the tree, it will have to visit over 10 to the 30 branches. So the only realistic option is to limit the number of predictions it's going to sample from and make a decision based on the information it can get from that. We needed to set that number for this prey, but we didn't know how much the prey needs to sample to be able to make a good decision. So we created an experiment. We incrementally increased the number of predictions we let the prey sample from until we see that the survival rate maxed out or a plateau. We started with 100K, which is a big number in computing power. And the behaviors we saw were very close to random. And the survival rate was very bad, very poor percent. And one of the things that we observed there is like it did not get better for many increments. Only got to a 8.4% survival rate after 100 million iterations, 100 million predictions. And we can only see from this graph, there's a little bit of improvement happening after 50 million predictions. So this is kind of like a strange situation because it looks like it's a super complex problem for a simulated prey, but we know that biology could deal with this with an incredible speed and with simplicity. Another interesting discovery from this experiment was when we explored the predictions that the prey was exploring while making a decision, most of the time it was spent exploring branches of the tree that had no opportunity whatsoever. So basically it was looping back and forward in open space in front of the predator, for example. Here's the second part of the talk, and I told you I was going to give you a biologically possible method to drive down the number of predictions and maintain results. But to illustrate to you better on how we got there, I have a quick game. Please play with me. You have to imagine you're a 12-year-old kid again, and it's very important that you put yourself in that role. You're a 12-year-old kid, otherwise it won't work. Now imagine that it's Halloween and you're trick-or-treating around the neighborhood, and at some point you make the wrong turn and end up at the gates of an abandoned factory, exactly like in a horror movie. You hear a noise, you turn around, and there is a big monster running towards you. You run to the factory and open the door, and this is what you see. Now, I only gave you 1 second to look at that picture, but if I ask you what features do you remember from that image, there will be many different answers, but two will remain very consistent across all of them. Two features will remain very consistent across all of them: the broken window and the columns. And it makes sense because these two features contain opportunities to exploit two behaviors we know are very useful while escaping from a predator: fleeing and hiding. So it makes sense that we're equipped to identify these features very quickly. But the question is how? Well, previous research in our lab found evidence that suggests that early mammals might have evolved a sense of connectedness of the space based on visual cues. This was very useful, you can imagine, as animals on land can see much farther than animals on water. Hence, prey needed to know if the space was occluded enough not to be seen from far by predators. Let's see how the connectedness of the factory we just saw looks like. This is a 2D representation of the factory. You can see the columns and the window. And we use eigencentrality to determine the connectedness of the space in the map. Eigencentrality doesn't only evaluate how well connected the cell is to its neighbors, but also how its neighbors are connected to their neighbors and so on. You can see from the image, some of the biggest fluctuations in eigencentrality happen around the window and the columns. So we process the map again. And this time, we focus in finding the cells with the highest variances in connectedness. You can see a group of cells showed up, this time surrounding the features where we think the opportunities are, the opportunities for fleeing or hiding. We call those points of interest. In theory, the prey could use those points of interest as a shortcut to detect opportunities around the column and the windows. Instead of expanding the tree in every step in every possible direction, we can make the prey expand the tree only when it gets to one of the points of interest and only in the direction of other points of interest that are visible from that cell. We think this can facilitate all the hiding and fleeing strategies to show up because we cannot force the planning to move in the directions of the features where we think the opportunities exist. Something else is it will prune the tree by cutting many of the useless behaviors and letting the prey focus on the specific actions that really affect the reward function. So the next step was obviously modify our previous experiment and introduce points of interest to our simulation and just repeat it. And we started with a smaller number this time. So we started only with 1,000 predictions that we let the prey visit before making a decision. And the results were astounding. With this minimal sample from the tree, the survival rate climbed to 3.4%. Remember, at 100,000, we used to have 0.01%. So it was very small. And he was able to, yes.
Could you take one more minute, please, so that.
Oh yes, I will wrap it up in 1 minute. And he was able, and also he was able to discover strategies using objects in the space to hide itself and make it to the goal. Let's move to the final slide. So we compared the results between these two methods, and we were able to reduce 99.99% of the sample of the tree that is necessary to create a good plan and maintaining similar outputs. This is still work in progress, but we believe we can enable with this technology an implementation for a fully independent robot with planning abilities that could successfully interact with an animal in real time. If you're interested in our work, you can play a game that we created in our lab that illustrates some of these concepts that is in the URL that is there. And this is all. Thank you so much for your time. And I'm sorry we had this mishap at the beginning.
No worries at all. I'm going to, since we don't have any Q&A right now, I'm going to encourage the viewers to put questions in the Q&A, but I have a quick question for you.
Yes.
So you know, your work is really interesting and, you know, so generalizable on beliefs about the environment. But then how animals also have like internal like uncertainties and beliefs. How do you think that would play with like uncertainties about the environment?
That is a great question. Yes. So the way that we compensate for uncertainty is by having to create more simulations. I'm talking from the computational biology perspective, right? So like we don't know exactly how that happens in biology. But the way that we compensate for uncertainty is by creating more simulations, for creating more possible values for the variables that we're interested in. So the only way to drive it down is by being more certain about the values or by knowing what are the possible values of those variables that we are not interested in or they will not affect the results. And I think that's where this research had some contribution on.
That's great. Thank you so much. So I'll applaud on behalf of everyone else. I'm going to introduce our next speaker now. Our next speaker is Professor Malcolm McIver, and he's going to talk about tuning movement to optimize information harvesting. He's a professor in mechanical engineering, biomedical engineering, and neurobiology at Northwestern University. So without further ado, I will pass it over to you.
Yeah, so I can't turn on share screen because it's giving me an error. There's somebody else sharing the screen. Herman, do you need to shut down screen sharing?
Herman, you can unshare your screen and also like turn off your video.
I think I did that. Let me just try that. Stop video. And I turned off. Yeah, I'm not sharing screen anymore.
I think we can still see someone's screen. Maybe on the top right, you should see a button called Stop Share.
There you go.
Okay.
Good. Okay, good. All right, so let me get this. Okay. Thanks, Needy, for the introduction. And I'm going to maybe go a little bit faster so that we can make up some time. Just to call out my collaborators, the PhD student who did this work is Chen Chen. He has left for the private sector a few months ago after graduating, so I'm going to give his talk essentially. And Todd Murphy is the faculty collaborator at Northwestern in mechanical engineering. So a lot of people have looked at this problem of how sensors move in animals, be they the pinnae of bats or whiskers or eye movements. One can ask this question as well from a more theoretical perspective of how sensors ought to move were it not for biological constraints. And here's one possible approach. You could estimate how much information a sensory organ can gain by placing that organ at any point in the space, and then generate a trajectory that at each step leads to an increase in that information, okay? And so that's what we're showing here with this rat following this gradient of odor to hopefully localize the source of the odor. So this algorithm has been published. It's Infotaxis back in 2007. There's a couple of issues with it. One is that if the information field is flat and say lower than your current position, then the algorithm would prescribe no movement at all, since there's no additional movement to be gained, no information to be gained by movement. So that's a problem because animals rarely stall out, if you will, in the absence of information. They're more likely to start exploring. And the other is that if there are distractors, real or fictive, it's easy to go to the wrong target. And so in fact, in this case, we have the actual targets over here, but because the local gradient led to a different sort of local maxima, the animal didn't get to that actual target. So our approach is called energy constrained proportional betting. And in this approach, what we do is we have trajectories that sample information space proportional to the density. And so in particular, in low density areas, you sample infrequently and move quickly, whereas in high density areas, you sample more often and move more slowly. And the important thing is that even in low areas, you still get some samples. And in particular, eventually you'll sample the area of the true target in this case. And so there's good distractor rejection. So the way the rest of the talk is going to work is I'm going to tell you a bit about some measurements we did on how sensors move in the context of electric fish, and then compare the measurements to the predictions of energy constrained proportional betting. The comparison to Infotaxis we leave for the paper we published in eLife last month. So if you're interested in that. So here's the behavior. It's really interesting. It's a natural behavior that the animals do in the wild, but we have here a plastic tube, and the animals try to hide in it as you move it fore and aft. What we think has happened in terms of its natural environment is these animals hide during the day in the aquatic vegetation of Amazonian rivers, and those are buffeted side to side by currents. And so the animal needs to stay with the weeds in order to be well hidden from predators. And at night, they come out and hunt themselves. So we have a big tank in the lab. We have a refuge underneath, or a refuge that is open on the bottom. We have a camera looking up, and we have a motor out of view here that is computer controlled to move the refuge forward and backward in exactly the trajectory we specify. And this is all under infrared illumination. Weakly electric fish emit an active electric field and are able to sense the location of objects by the way that object distorts their field. And what we are able to do is we have a knob on the signal to noise ratio. Through work that has been done over the past decades, we know that by injecting an electric field that is similar to the animal's own electric field, we can jam the electrolocation system. And since we can carefully control that, we're able to smoothly vary the signal to noise ratio from 0 to some large value where the system isn't working very well at all for the animal. So here's some data. Here is the refuge position in blue. Here is the animal's position in green. And this is in the strong signal condition with no jamming. Once we put some jamming in, the animal starts doing these fore-aft body oscillations to better localize the refuge with its electrolocation system. And we quantify that change in the amount of movement by something we call relative exploration, which varies from unity when it is the exact same amount of movement as the refuge to some larger number. So it's the length of the movement taken divided by the length of the movement taken by the refuge. And that increases from strong to weak signal conditions. And we also do spectral analyses, and I'll just call out the fact that in this zone away from the movement frequency peak of the refuge, which is at 0.1 hertz here, the amount of movement in the higher frequency domain for the weak signal condition increases, okay? So we have a complicated algorithm I don't have much time to go into, but essentially just jumping into it quickly, the algorithm computes an estimate for the location of the refuge, which is the belief. It computes an expected information density that has this characteristic double peak shape for a belief of this shape. And then where our real contribution lies is in generating a trajectory that proportionally samples this, this information distribution balanced by the kinetic energy and needed to execute the trajectory. Okay, so it's a careful balancing. And what that will predict then is that you'll have to sort of sample in this area more, but still sample a little bit in that area. And so if you look at the sampling density here, derived from this computed trajectory segment, you see that the sampling density is what it needs to be. And then it comes back, executes the trajectory, takes some measurements, integrates it with the prior belief, and computes a posterior using a recursive Bayesian filter. Okay, so underneath the hood is a functional that essentially has one term that is information and one term that is energy. And this term uses a metric called the ergodic metric, whose value is 0 if you're proportionally sampling the distribution perfectly, and goes up from there as you do more imperfect proportional betting. And then this term is simply the kinetic energy needed to carry out the trajectory. Okay, so you could try to go for perfect proportional betting, but that's going to require a lot of kinetic energy. And on the other side, you could go for very little kinetic energy, but that's going to lead to poor proportional sampling. So this functional sort of balances those two terms. And what happens when we do the simulations, here's the original data, and here's an example of what our simulation does in the strong signal condition and in the weak signal condition. All right, so here's some statistics for the measurements across all trials. In the strong signal condition, low relative exploration, and then it goes up with weak signal conditions. Here's a representative trial, and here's the statistics over a bunch of those trials in terms of showing that with weak signals, we have more energy or more movement in the higher frequency domains. All right, similarly, the model duplicates or replicates this result or predicts it, depending on your perspective, including the increased investment in high frequency components of movement, also shown by this plot here, where we collect statistics over many trials of simulation. All right, so we wanted to, get out how is it that these sensor oscillations that are observed in many different systems do they actually lead causally to better performance? So our approach to looking at that is to take the original movement trajectory prescribed by the algorithm, which is shown in orange here, and then progressively attenuate it, and then execute that trajectory and ask how much our performance, our localization performance is affected. Okay, and that's shown here. Our mean relative tracking error goes up as the amount of those body wiggles, the amount that we attenuate those body wiggles goes up. So you see a nice relationship there. And we can also, one of the things I'm very excited about with this data is we can look at, this weak signal behavior of oscillating the body a lot is energetically expensive. And spending energy means you have to hunt for more prey, which is a dangerous endeavor. So why is it doing this? Well, we can see this is the data that shows the additional energy needed. And we can see that, again, the relative tracking error goes up quite a bit as we do fewer and fewer of those wiggles, okay, in the weak signal condition. And also we can look at the mean tracking error against movement attenuation here. And what I wanted to show you is mean distance from ergodicity versus the amount of attenuation of those wiggles. So as we attenuate the wiggles more, we're further away from proportional bedding, the ideal proportional bedding scenario. And then we can look at distance from ergodicity versus tracking error and see that they're nicely related as well. Okay, we've done this analysis on several other species shown here. I can't discuss them for time, but I encourage you to look at our eLife paper published last month where we describe this. And in summary, EIH generates good qualitative agreement with measured trajectories in strong and weak signal conditions. Animals appear to gamble energy for the chance to make an informative observation. And we'd like to propose that ergodic movement is an embodied component of information processing. And with that, thank you for the opportunity, and I'd be happy to take any questions if there's time.
Thanks for your talk, Malcolm. I'll applaud on behalf of everyone else.
Thank you.
Since we don't have Q&A right now, let me ask you a question. That was a really interesting talk, first of all, and I think the way to balance out information and energy or try to have them both show up in the same objective function is really interesting. I'm interested in how you chose to, add information and energy in the same objective function, given that you don't really think of them as having a common currency, or is that where your cost of information shows up? Like your lambda, basically. How do you weigh
Right, yeah, so the lambda, those prefactors are essentially things to get the units to work out. That's correct, yeah. Well, but awesome. Yeah, these are, the ergodicity metric is 0 to some large number as you go away from proportional bedding, but it's a non-scaled, it's a unitless value, whereas the energy is kinetic energy. So yeah, there's some unit work there to make things pair up properly. Not sure if that's your question though.
Yeah, that was my question.
Oh, okay.
Yeah. Thank you very much. Since we're a little short on time, if people add questions for you in the Q&A, please feel free to type your answer in
Certainly.
there.
Thanks very much. I'll stop.
I
think I've stopped sharing.
Introduce our next speaker. Thank you. So our next speaker is Monica Yoja. She's going to talk about synapse size as a gate between stable and switch-like behavior. Her work applies control theory to neuroscience, and she's a postdoc with Timothy O'Leary at University of Cambridge. I'll pass it over to you, Monica.
Thank you very much, Liddy. I will share my screen. Can you see it?
Yes, we can.
Okay. Great. So today I'm going to talk about an idea that my supervisor and our group leader, Timothy O'Leary, had some time ago, which is that neurons might use their synapse sizes to elaborate different types of computations. So synaptic potentiation means that after spiking activity, synapses grow. And let's focus on the dendritic spines of postsynaptic neuron. And So when this growth happens, it happens on a much longer time scale than the spike itself. So for this to happen, you need a switch mechanism inside the synapse, which happens through many complicated chemical reactions. And one that's proven to be necessary for it is the autophosphorylation of COMK2. So what happens here, this is a complicated picture, and I won't go through what happens because this is not the focus of my talk. But what happens is that there's a spike coming in, and this enzyme called COMK2 gets activated. And when the spike is already gone, it's still active for some time. So it keeps this signal on. And there is another thing called, it's a phosphatase called PP1, which deactivates slowly this COMK2. So this is how a switch mechanism can happen. But I will get back to it in the end of my talk. So for now, let's take these COMK2s, more precisely the active COMK2s, and free PP1s. So those PP1s that are not busy deactivating COMK2s. And if you take these two populations, then we may notice that they inhibit each other. So we had this more general question, which is, how does a system where there are two mutually inhibiting species present, how does the system behave? And more interestingly, how does it behave when there is a small number of molecules present and when there is a large number of molecules present? So we took this simple mathematical model called toggle switch, where you can think about X as an on mode and Y as an off mode. So these two, let's say, chemical species inhibit each other through these rates, these hyperbolic rates. And they have a linear death rate. And what's important here is this X bar and Y bar, which is going to be, these are just constant parameters, but they will give us the average number of molecules present in the system. And for the time being, we will assume that X bar equals Y bar, and we'll call it the size of the system. So X is like the number of molecules from species 1, and Y is the number of molecules from species 2. So if we simulate this process by setting X bar, Y bar equal 2 and X bar, Y bar equal 100, then we use Gillespie algorithm. And then what we see is that we get this time series. And what's important here is that on the left-hand side, you see this process bumping into the x-axis several times. And this doesn't happen on the right-hand side. So the right-hand side is like a random walk with some attraction line level. And this one on the left is a little bit different. So if we plot the distribution of these processes, then what we can see, as we might expect, is that the one on the right looks a little bit like a Gaussian distribution, but the one on the left is very different. We have some bumps on the axis, and then we have, in this case, a third little bump in the middle. So we will call these modes. And so this system acts a little bit like it acts like a switch. It has these two big bumps, and it spends a big amount of time in each of these. And this other one behaves like a stable system. It is always around its one mode. So that was basically our answer for the first question through this simple example. But in reality, it's really hard to, sometimes it's hard to simulate actual biochemical systems. So it's good to have an analytical tool to at least have a guess what you will get, if it's going to be a bimodal or multimodal system, or it's going to be a more stable like unimodal system. And with this purpose, we applied linear noise approximation for this toggle switch model, which just means that we approximate these processes as like a noise process that a noise process around this equilibrium, this average number of modes. And if you do that, then you get Gaussian processes, so like a Gaussian fit around these points. And what's interesting there is that you can see that the probability that goes beyond the axis, so beyond the region of interest, beyond this positive quadrant, is very large in case of multimodality, and it's very small in case of large systems or unimodality. So we wanted to check this for like a more general class of systems. So for this reason, we don't assume that x bar equals y bar. And also we had a few more extra parameters, k1, 2, 3, 4, instead of just fixing it as the same parameter. And so if we had that, we randomly generated constants. And so every time you have a set of k's and x bar, y bars, you get a blue circle here. So what this plot says is that this probability mass outside the axis correlates with multimodality in this class of systems. So going back to the active COMK2 and free PP1 system, when we again have two mutually inhibitory populations, then what we can imagine happening is that at rest, there's not much active COMK2s, but there's a lot of free PP1s. And when there is a spike coming in, then all of a sudden, many COMK2s get activated, and PP1 starts working on deactivating these COMK2s. But there's not enough, in the small system case, there's not enough PP1, so it gets saturated. And due to that, we get another mode. So for some time, the system gets stuck in this on mode, which causes growth of the spine. And then after some time, it goes back to this resting state. But this phenomenon doesn't happen when you have enough free PP1s and a large number of COMK2s. So just to sum up, so we talked about that depending on the system size, we can get switch-like behavior or stable behavior. And we talked about two different systems, but similar in a way. They are both like mutually inhibitory systems of two populations. One is a more biological example, is COMK2 and PP1. The other one is more mathematical model, which is the toggle switch. And we showed we proposed that this switch-like behavior and stable behavior might be used in dendritic spines. So at last, we also talked about an analytical tool to foresee if a system becomes multimodal or not with the linearization of the system and with this overspilled probability mass. So the probability mass that goes beyond the axis. But the bigger question is basically that does biology use this tool, this computational tool, that by changing the size of a system, it can change the core computation, the type of computation that happens within it. And with that, I would like to thank you all for listening.
Thank you, Monica. I think we have a question for you. So the question is from Marcel. He asks, he thanks you for your talk, and he asks, what do you think your findings indicate for heterosynaptic plasticity? Does that mean that a small synapse might be stabilized by big neighboring synapses due to diffusion of PP1 molecules from the big synapse to the smaller one?
That's a very interesting question. So I thought about like supply more through the dendrites, but it's a very which actually can happen with neighboring spines. I didn't actually think about it or use it in the model, but I think it can happen, yes, that neighboring spines stabilize or deactivate this on state.
So Marcel, does that answer your question? Hopefully that answers your question. Actually, since we'll get kicked out of this room in 5 minutes, but since we had to, skip over the other two speakers, if the other two speakers want to take a few more questions, like the questions that they answered in the chat, if they just want to answer it live, that would be cool. Yeah, since we have 5 more minutes. Otherwise, I can just summarize the Q&A. So one question that Herman got was, can you speculate what the survival rate, why the survival rate is limited with 10%? Is there a way to increase it further?
Yeah.
And
oh, sorry.
Yeah. Herman, is that you? Go ahead and you can share your video and answer the question if you like.
Thank you. So yes, and the answer was related to the scenario that I showed was one of the many scenarios that participated in that experiment, and you have to remember the predator is way faster than the prey. So that means like if there is a way to chase it to catch it, it will do it. And also in most of these scenarios, the spawn locations are randomized. So that means that the predator will be located between the prey and the goal. So those scenarios are barely survivable. It will require a good plan plus a lot of luck to make it out.
Cool, thank you. And we had one more question for Malcolm. It was really cool model. Is there a way to experimentally measure information gain in a direct way? And they've been having a conversation about it in the Q&A. Malcolm, do you want to answer that question live before we close?
Sure. It's unfortunately not much to answer. It's a great question. I wish I could say, I mean, even in the places where we've done some really detailed work looking at looming within larval zebrafish and changing things like approach rate and that sort of thing, it's clear that what we call belief in our model is distributed somewhere between the retinal ganglion cells in the ventral temporal part of the retina, somewhere between there and the reticular neurons that are doing some of the processing downstream. But beyond that, it's hard to know where to look and hard to know how to quantify belief even in that very simple case. So I don't know how to measure the belief directly, unfortunately, but I'd love to get some input on that if people have ideas.
Great, thanks for walking us through your thought process there. Cool. So thanks to all the speakers and all the attendees for the awesome session. Since there are no further questions, I think we will close now. But I will applaud one last time for all of you. Thank you.
Thank you very much. Bye-bye.
Bye.
Throughout the session.
Mm-hmm.
And then in the chat, there will be some questions in the chat as well. Please check the chat as well, especially after your talk. There will be some questions for you and some comments about your talk. It's always good to check it. And we are currently using Zoom as well as YouTube. The participant number you are seeing in here is not limited. We have YouTube record, some people from YouTube. We won't be able to see how many people are watching there. And we will be recording this video. If you have unpublished data which you don't want to be shared later on in YouTube, please let us know so that we can cut this part. If you want to just share it online now, but not in the future, because we are recording and they will be available in YouTube as a, recorded videos. Let us know if you don't want to put it for later on. And let me see, there's one more point I need to wish. Yeah, in the chat, you can either directly contact me or Zoom 8. I think for us, Melvin is with us, although we don't see him. He's this room 8. Melvin is going to help us for technical issues. If you have any problems, please go directly and ask him through the chat during the session. He will fix it for us. And I think that's it. Do you have any more questions?
No. Do you mind if we have questions for the other speakers? Do you mind if we just speak directly?
I prefer to write it in the chat, and then you can go through it, because we, first we our priority is to ask the questions from audience and then you. Sure. But if we have more time, yeah, we should go for it. I would be happy.
Yeah, I mean, if for example there's no questions or.
Yep. You should go for it and unmute yourself and feel free to ask your questions, because you will be able to see the questions in the question and answer panel. Yeah. If there's none, please go for it.
All right, cool.
And because the relaxed you are, the session will be much nicer. And please be more interactive, just like writing in the chat or throughout the panel, question and answer panel. The more interactive you are, the more interactive the audience will be. So it will be much nicer.
Yeah, it's also cool, I think,
we have some audience currently who is watching us.
Sorry?
We have some audience, I think, who is already started to watch us.
Oh, cool. No, I was as a suggestion, like if we speakers keep the camera on while the other speakers present, it's kind of nicer to see.
When you are presenting, you will be the only one. You will be able to see us. As panelists, you will be able to see the other panelists. But people who is watching it online, they will only see you. And the other thing is for timing, I will use virtual background colors. For example, for your last minutes, I will use this card for you. And for 2 minutes, I will use this. But again, as I said, this will be available to you, but not to the audience. And I hope you will start, you will finish around 12 minutes. I know it is a little bit, when you see the red card, I know people start, talking quicker, but yeah, don't worry about it. I don't want you to stress you out. It's just like a reminder to you. But if you finish in time, we will have more questions and more interaction. We have 3 minutes and we will wait for more participants. And for our participants,
I'm just going to quickly plug into LAN just in case my internet screws up.
That sounds good. For our participants, if you want to ask any questions like details, everything, feel free to ask them throughout question and answer panel, as well as chat. Because we have very limited time for questions, I will choose the questions which is voted the most. That's why it is also important to vote the questions in the question and answer panel. And I will choose the questions from question and answer panel more than the questions in the chat. That's why you should use question and answer panel to ask the questions first and then vote them.
Great.
Okay, we have last 2 minutes to go.
Yeah, thank you very much, Huriye, for hosting the session.
We are very thankful to you, sharing your talk and work with us. It's beautiful. Okay, we are waiting for 2 more minutes to start our sessions. For other participants to join us, then we will go and we will start our session.
Cool.
Okay, this is last minute before we go online. Hello, everyone. Thank you for joining us in this session. We have 3 amazing speakers in this session. Please post your questions in the question and answer panel down in the webinar you will see. We only have few minutes for questions, very limited time. That's why we will choose only 1 or 2 questions to answer online. Vote for your favorite questions in the question and answer panel, and those will be asked first. You can also use chat to ask the questions, but the questions in the question and answer panel will be prioritized. For the rest of the questions, our panelists will be around to answer them in the chat and the question and answer panel. Our first speaker is Anna-Sophie Lorin. She's doing her PhD at University of Montreal under the supervision of Arlene Kahn. Today, she will tell us her work on the mechanisms that might explain how we can perceive the world as stable despite our constant eye movements. And her recent work from Kahn Lab will be shared in the chat shortly. Stage is yours, Anna.
Thank you. Thank you so much for being here today. I'll share my screen. Okay, so in this presentation, I'll explore the role of predictive mechanisms and more specifically, pre-saccadic attention in perceptual stability across saccades. So despite our impressions, we only see the world in small snapshots, with the center in high definition and in color, with all the surroundings blurred and dark. So we must constantly move our eyes from one point to another and then to another to encompass the whole scene. And this is done with very fast eye movements that we call saccades. And this causes images on the retina to move constantly. And yet we still perceive the world as stable, and we believe we can see entire scenes at a glance. So this paradox is called perceptual stability. And the question remains, how is it achieved? So it has been suggested that it might actually rely on predictive mechanisms. So before we move our eyes, we make predictions about the view after the saccade. And then after we move the eyes, we compare visual information obtained before and after the saccade. To do that, we must first displace our covered visual attention in the periphery to acquire information and hold it in our memory for future comparison. This facilitates perception and is called pre-saccadic attentional facilitation, where discrimination at the peripheral goal location of a saccade is enhanced. So now, the question is, does pre-saccadic attention help perceptual stability? If so, we presume that its role would be to make accurate predictions about our perception once the saccade is made. So we explored this in two main experiments, but first we had to have a measure of pre-saccadic attentional facilitation. So to do that, we implemented a dual task paradigm with an eye tracker, which recorded participants' gaze. So first, they were fixating a dot surrounded by 6 figure 8, as you can see on the right, on the left. And then we asked them to make a saccade to one of the figures as soon as the dot would turn into an arrow, indicating which figure to look at. And then the second goal was to discriminate between 4 letters called the discrimination symbol. And it would appear for a very, very short time just before the saccade, right here. And if the task worked, it's only because participants' pre-saccadic attention was already directed to the target that they could successfully discriminate the letter. Their pre-saccadic attentional facilitation would allow them to perceive the target's identity. But that would be the case only in 50% of the trial, where the discrimination symbol would appear actually at the saccadic goal. In the other 50% of the time, it would appear elsewhere to any other placeholder. So first, we only compared participants' discrimination performance between the valid position, where the letter to discriminate appeared at the saccadic goal, and invalid positions, where it appeared elsewhere. So we can see here that participants were much better when the discrimination symbol was at the saccadic goal. This confirms that our task really measures pre-saccadic attentional facilitation. We can distinguish a target much better if we're about to saccade to it. We can also see that performance is at chance level in invalid positions. So we'll be more interested in valid conditions from now on. So anyway, this solid effect replicates previous findings on pre-saccadic attentional facilitation. So now that we are sure we are manipulating the right variable, the next goal was to determine if changes occurring after a saccade could influence pre-saccadic attention. So what happens after a saccade should be irrelevant to a pre-saccadic task. If it's not, we would assume that pre-saccadic attention is involved in the comparison between the pre- and post-saccadic perception. So whether we retain pre-saccadic information or not would depend on the correspondence between pre- and post-saccadic images, indicating a stable world. So to resume, if pre-saccadic attention is involved in comparing pre- and post-saccadic information, a change in a post-saccadic change should disrupt these comparisons, which in turn should disrupt pre-saccadic attention. On the other hand, if pre-saccadic attention is not involved in these comparisons, post-saccadic changes are not relevant and should not change anything. So we measured pre-saccadic attention again, but we had 4 different conditions to induce post-saccadic changes. So in the baseline, nothing changed after the saccade, and that constituted our comparison standard, which should yield the highest discrimination performance. Then in the all-off condition, all possible placeholders disappeared right after the saccade. Next, in the one-off condition, only the discrimination symbol disappeared, which was also the saccadic goal when it was in the valid position. And finally, in the one-on condition, it was the opposite. So only the discrimination symbol remained after the saccade, and all other placeholders disappeared. So then we compared discrimination performance across all conditions. And we saw that the one-off condition, where only the discrimination symbol disappeared while every other placeholder remained, was associated with a lower performance, not only than the baseline, but also all other conditions. And we did not see any difference between any other conditions and baseline. So this was a little surprising for the all-off condition because the discrimination symbol also disappeared here. But to explain this result, we assume that in this case, the situation was more comparable to a blink. So if there is no comparison to make between pre- and post-saccadic scenery, there is no judgment to make about perceptual stability. So we saw that pre-saccadic discrimination performance was only disrupted when its target disappeared, but without its surroundings. So it was expected that its disappearance should impact performance. But as we saw with other conditions, these findings are inconsistent with an explanation of a large change in the scenery disrupting memory, but rather they suggest that changing a saccade's specific target location can disrupt pre-saccadic expectations about said target. So this brings us to experiment 2, where we shifted the target instead of causing it to disappear. So along the same logic, a shifted target should impact performance because it does not coincide with the predicted expectation of its location before the saccade. However, saccades are noisy. They do not always land exactly where we want them to. And more importantly, our brain is perfectly aware of this variability and take it into account. So saccade saccade variability should also play a role as well, with discrimination symbol shifts disrupting performance, but only when shifted outside of the normal saccade variability. So in this experiment, we had two main conditions. In the first, the discrimination symbol was shifted parallel to the saccade from 3 degrees inwards to 3 degrees outwards. And we have an example here. And in the second condition, the discrimination symbol was shifted perpendicular to the saccade from 20 degrees counterclockwise to 20 degrees clockwise. And we compared these performances between baseline in these conditions, while also taking into account saccade variability, looking at participants' saccade landing positions. So first, we looked at the distribution of participants' saccade start and end positions. And we can see that there is variability around the figures, and it did not change between the conditions. So in previous studies, there was evidence of more variability parallel to saccade vector compared to perpendicular, but not so much for oblique targets where evidence is less clear. And indeed, we can see here that it forms an elliptic region parallel to the saccade, but it's less pronounced in oblique position, even though we can still see it a little. So next, we compared performance for every parallel discrimination symbol shift. Of course, the 0 degree shift corresponds to baseline. And you can see on the right that we also compared the participants' distribution of their saccade amplitudes to the target's actual position along with their performance. So first, we saw that performance was lower when the discrimination symbol was shifted 3 degrees inwards and for all its outward shifts. On the right, we can see that the saccades were slightly undershooted in general. The baseline corresponds to the target's initial position at 5.8 degrees from fixation point, and the majority of saccade landed slightly inwards with the mean at 5.4 degrees from fixation. We can also see that a higher proportion of saccades landed 1 degree inwards compared to 1 degree outwards. So this could explain the performance results that we have here. Next, we looked at the same parameters, but with perpendicular target shifts. And this time, we saw a performance decrease for all target shifts compared to baseline. And this is understandable because, as I said before, saccadic variability formed an elliptic distribution parallel to the saccade. And perpendicular shifts, as we see on the right, were much more likely to fall outside the participants' saccade variability ranges. So to resume this experiment, we saw that pre-saccadic discrimination performance decreased with target shifts in a similar manner to their disappearance. We also observed that their performance was linked to their saccade variability because small shifts landing into their endpoint range was associated with better performance compared to bigger shifts. So in conclusion, in experiment 1, we saw that only changes to the saccadic goal where pre-saccadic attention was directed disrupted performance, but only for condition where every other placeholder remained. And in experiment 2, we saw that performance was disrupted by most target shifts and was linked to saccade variability. So these results lead us to conclude that pre-saccadic information is more likely to be disrupted when we perceive a change in a target's position, but within the same scene. So these findings suggest that pre-saccadic attentional facilitation indeed plays a role in predictive mechanisms for perceptual stability. So I want to thank all of my collaborators for this project and thank you for your attention throughout this presentation. And of course, I welcome any questions that you might have. Thank you.
Thank you, Anna-Sophie. This is a beautiful talk. And for our audience, please ask your questions in the question and answer panel for Anna for the details or any discussions she would like to you want her to answer. We have one question for you, and Megan Peters is asking, could the differences, if you click the question and answer down, and you will be able to see it, could the difference between everything disappearing and only the target disappearing have anything to do with the initial encoding in the periphery being subject to crowding and thus the creations of interference? This is not about from questions yet. Yeah.
Just being subject to crowding. Well, we saw with different conditions, initially the information pre-saccadic attention with the information acquired with pre-saccadic attention was well encoded. And then the difference between everything disappearing and just the placeholder disappearing, we expected to be since it's a larger change, a much bigger change when everything disappeared, we expected that it would disrupt performance even more if it was like just a memory thing. But it's actually the opposite. So yeah, perceptually, we perceive less of a change when only the discrimination symbol disappears. So I hope it answered the question. It's not like, surely it can't be this, but yeah, it's our...
She is basically wondering the interaction with the iconic memory and the crowding. Maybe, you know, the interaction between these two things is just like appearing some change in the people, you know, saccade dynamics. That's why, you know, wanted to ask and clarify.
Yeah, we just weren't interpreting that changing only the discrimination symbol would create more crowding, but yeah, it's possible.
Okay, thank you. We have another question for you from Priyanka. Priyanka says it's a great talk. Thank you. Have you looked at performance as a function of time of change offset relative to saccade onset? Do you see any signatures of the saccadic suppression?
Well, performance as a function of time change offset. Well, everything was made very quickly, so it's the participant saccade that that would create the change. So as soon as the participants started their saccade, and this was calculated with a velocity criterion. So yeah, so as soon as they made a saccade, the scenery would change. So the change offset, I'm not really sure what you mean by the change offset.
Priyanka, could you please clarify your question for us? Yep. Do you think this uncertainty might be, you know, you are changing two things in the stimuli, and maybe this uncertainty affects it one more than other for these features? And what we see in this case, I don't know, I'm just like speculating because you have interesting results. Yeah. Do you think it is related to the uncertainty in the stimuli?
Sorry, could you repeat the question?
I mean, yeah, I'm just wondering, you know, you are playing with different aspects of the stimuli, and then one is has stronger effect than the other. I'm just wondering maybe that these features of the stimuli affecting the saccade more than others because it causes more uncertainty for participants. You see what I mean? But I'm just like wondering, can it be due to the correlation with the uncertainty in the stimuli?
Yeah, it can be that too, because yeah, it's true that with everything else remaining the same, it can create more confusion. So
thank you, Anna. Now feel free to ask more questions to Anna. She's around throughout the sessions, and she will be happy to answer your questions in the question and answer panel. Thank you. It is a lovely talk and beautiful results.
Thank you.
Now we will move to our next speaker, Alex Hernandez Garcia. He has just finished his PhD at the Institute of Cognitive Science of the University of Osnabrück in Germany with Professor Peter Koenig. Today, he will present the results of an eye tracking experiment in which whether some images are more likely than others to attract our visual attention. The work is recently published, and the link will be shared in the chat shortly. Alex, screen is yours. I don't see Alex.
No, okay. Now you can hear me, right? And you can see my screen.
I can. Yep.
Cool. Yeah. Thank you very much, Lidia, for the introduction, and I want to thank the organizers of Neuromatch as well. So I'm going to talk today about global visual salience of competing stimuli. I'm going to try to move this thing. Ooh. Well, sorry, it's not working for some reason. My Zoom. Anyway, I'm going to talk about global visual salience of competing stimuli. This is work with Ricardo Ramos Gameiro and Peter Koenig in Osnabrück and Alessandro Glini from Groningen. So to start with some motivation, well, so ultimately we want to understand human visual attention. I have this here. Of course, we have more important questions for humankind. But this is to say that we are here interested in understanding human visual attention. And there are so many approaches to this question. One has been computational models of visual saliency. And this has been very popular. And for example, recently, Matthias Kummer et al. Proposed deep gaze, which computes very accurately the distribution of fixations on natural images. So you get something like this. If we have another image, it would be something like this, like a salience map of a natural image. But here we ask, so what if several images compete for our visual attention? And we question, okay, are some images as a whole more likely to attract human visual attention? And in other words, we are interested in finding out if some images have, or if natural images have an intrinsic global visual salience. So an outline of the talk would be, and here I'm bracing a spoiler alert because I'm going to tell you like the findings, and I will try to convince you through the presentation. So first I will briefly talk about the data collection, which was an eye tracking experiment. Then I will present the computational model where we compute the global salience of images, and then straight away to the results. There are a few of them, but I'm going to focus today on 3 of them in the interest of time. So about the data collection, we performed an eye tracking experiment where we had like 200 natural images from 6 different categories, almost 49 participants, and we had 200 trial per subjects. And in some trials, we varied a small task the participants had to make. We also mixed new with old images to get the variability to study these factors. And then we modeled the behavioral data from the participants with a computational model, a logistic regression model. And I don't have time to go into all the details. You can find this in the paper, but essentially the important bit is that we modeled this with a design matrix where each row corresponds to a trial. And we have, this is like a vector where we have 200 entries in this vector, 200 numbers, which are because we have 200 images. And 2 of these elements in the vector will be the image that is presented on the left and the image that is presented on the right. And all other elements in the row are 0, and the left image will be 1, the right image will be -1. So in this case, we are giving our model the information about that trial, which images were presented on the left and on the right. And we modeled this with logistic regression, and in particular, because we have pairwise comparisons, the model simplifies, and actually recommend a lot using this for pairwise comparisons because it's really handy. And we have here our design matrix, and because we wanted to account for other factors like task, familiarity, and potential lateral biases, we added elements to the columns. And again, I will not go into the details, but we have elements that accounted for the task, for the familiarity, and for the subject biases. So we trained this logistic regression model to predict which image, left or right, was fixated first. And the model actually performed very well. This is good news. So for example, it predicted which image was fixated first with about 82% accuracy, 82% accuracy with a high error under the curve. And this is not the main results. This is just to tell us that the data, the behavioral data, could be predicted and learned by a computational model, a machine learning model. And that means that we can use further the coefficients that the model learned. So now about the results, the main result of our work, of this paper, comes from looking at the coefficients of the images, the coefficients of the model. And as I said before, there is 1 coefficient per image. So that this allows us to rank, for example, the coefficients of the images. And we observed that images that contained faces, like close-up faces or humans, obtain the highest coefficients in the model. And after that, images of natural and urban landscapes came. This is another way of looking at this. As you can see in purple on top, it's data points that correspond to faces, humans, and these get the highest coefficients. So we called these coefficients, this is what we describe as global salience, because this somehow models the likelihood of every image to be fixated first when it is presented next to another stimuli. And we might be wondering, okay, this is cool, Alex, but this might be just explained by the local salience properties of the image, and we also wonder this, and we wanted to account for this. Or is this a new an independent measure, or is something that that could be explained by global salience? So to test this, we computed the salience maps with two computational models. One is GBVS, a good old GBVS, and the other one is DeepGaze-2, more modern, more accurate. And we perform two types of analysis. One is to check the predictivity of salience maps for predicting the location of the first fixation. And the other one is to see if these salience maps could predict, could correlate with the global salience scores. So here are the results. We computed the KL divergence between the fixations maps of the first fixation maps and the salience maps of the computational models. And we observed, this is for GBVS, this is for DeepGaze, we observed that it's larger than 0. It would, if it was 0, it would mean that the first fixations corresponds to the high salience areas of the computational models, but that is not the case. So that means that salience maps were not predictive for the location of the first fixations. This was one test. We performed other tests to explain the relationship with local salience. So we looked at the correlation between the KL divergence, this KL divergence between first fixation maps and salience maps, with the global salience scores that we computed before with the logistic regression model. And again, we saw that the correlation was either close to 0 in the case of GBVS, or slightly correlated in the case of DeepGaze. And finally, we looked at trial-by-trial comparisons, where we computed the salience map of the whole stimuli that the participants saw in the lab, so the two images at the same time, and we computed the mass of each side, the mass on the left and the mass on the right. And we again compared this with the global salience to see if it could explain our global salience scores. But again, in this case, that was not the case, and this is telling us that local salience properties don't account for our global salience scores computed from learning the behavioral data of our tracking experiments. Finally, for today, we looked at the lateral bias. So this has been observed before, that there is a tendency, at least for people who learn to read from left to right, and there's an open question. We also observed in our data that participants looked more often first on the left image than on the right. And our model captured this, and also we could look at this on our data. And this is also an interesting observation because it confirms previous results. Because we introduced coefficients in the computational model, we could also compare these coefficients with the percentage of fixations on the right image, and we observed that the correlation was almost perfect, which means that the model was capturing this well, and that means as well then the coefficients that we call global salience are more reliable. As a summary, we performed an eye tracking experiment with competing visual stimuli, pairs of images, and we studied the effect of task and familiarity, which I haven't presented today, but you're welcome to look at in the paper or just ask questions because I have some slides. And we also looked at the lateral bias. And we trained a machine learning model with the eye tracking data to obtain global salience scores. And we compared these salience scores with local salience properties of the images. And we found that local salience properties don't account for the global salience of natural images. And the main conclusion is that it is possible to measure the global salience of natural images that is independent of the local saliency, and that there is strong spatial bias towards the left side. And as brief comment about the results that I haven't presented. We found that task and familiarity play little role in explaining global salience and in this kind of behavioral data, and also that images with larger global saliency were explored for a longer time. If you want to know more, we recently published this at the Journal of Vision. There's also a preprint, and all the data and code is available on GitHub. And if you have questions, I'm very happy to take them. Thank you very much for listening.
Thank you, Alex. This is very nice results. We have one question from our audience. Muhammad Abdelhak is asking, does lateral bias correspond to conjecture of the dominant eye?
Okay, that's a good question. I don't have the data to respond to this question at the moment, unfortunately. I think it doesn't, because just from memory, there was a majority of right-handed participants, and there is this bias to the left. And also I can say that even if there is this strong lateral bias to the left image, there is a huge variability across participants, and that's why we modeled the bias of each participant separately.
Okay, thank you. We have another question for you from Tasvi Açlar, asking, what about comparison between similarity between the two images? For example, similar to pop-out, presumably if one has a unique features over the other, this can pull attention to one. I'm not sure the local or global salience captures this.
Mm-hmm. Yeah, that's a good point as well, and that's something we are looking at as a follow-up at the moment. So trying to look more closely at the particular properties of images. I could say that local salience would capture this pretty well, because they respond highly for this kind of pop-outs effects. And in general, what we saw with global salience is that because we are taking into account only the first saccade, the first fixation, then global salience would not, in principle, it would be more related to these sort of effects. That's why it was surprising that then we found that just images that contain humans or faces are the ones that receive the higher global saliency, meaning that they are fixated more often. So in sum, it is not so much related to low-level features of the images, but the social content, the semantics of the images have play a big role, meaning that in the periphery, before making the first saccade, we are able to scan the images and make this perceptual decision.
Yeah, having a cute baby images might change quite a lot.
Or cats.
Thank you very much, Alex, for your nice talk. Now we will move on to our next speaker. Our next speaker is Radhan. Just a second. Radha is doing her PhD at the University of Kayseri-Slautern, working in a number of areas such as visual search, analytic, holistic processing, eye movements, cognitive load, and handwriting research. Her primary area of interest are eye movements, scan pad, and working memory. Today, she will present her PhD work, which is recently published with Professor Cees Van Leeuwenhoek and Dr. Andriy Nikolaev at the Laboratory of Perceptual Dynamics, Brain and Cognitive Unit in Leuven. The link will be posted shortly in the chat. And stage is yours, Radhan. We can't hear you. Please unmute yourself, or I can unmute.
Yeah, I'm trying to.
Thank you.
All right. Thank you. Thank you for this opportunity to NeuroMatch as well. And thank you for the organization. Today I'm going to be presenting some work that I did during my PhD at the Perceptual Dynamics Laboratory with Professor Case and Dr. Andre. So this work is part of a larger effort to study EEG during working memory tasks under free viewing conditions. So why free viewing? Why is free viewing important or different from traditional working memory tasks? I'm not able to control this. Yes. Because eye movements, whenever there's free viewing and we have unrestricted vision, we tend to make eye movements and a lot of saccades. And what this brings is a lot of additional complexity, which are outside the purview of the experimental conditions that we are interested in. So as Anne just presented, what we know from quite some time is that when a saccade is planned, attention shifts to the subsequent fixation location before the saccade is executed. And this attention shift, turns out, is affected by a number of top-down processes, such as working memory, prior knowledge, and other task-related conditions. Another thing that happens when you talk about free viewing is this, refixations, which is essentially moving our eyes to a previously fixated location or area of interest. Now, this behavior is also common, and it has been observed in a number of different tasks, such as reading, visual search, scene viewing, pattern copying, and so on. And so this is a question that we were interested in. How does task-related refixation behavior affect saccade planning? And the highlight is that we were trying to do this under free viewing conditions. So to test this, this is the display we used, which is a visual search display, and with target T's and distractor L's. And as I told you before, what we are primarily interested in was a working memory task. So we had 3, 4, or 5 target T's among distractor L's, and this was our experiment setup. So participants were asked to look for these targets during the 10-second search. And what they had to do was remember the orientations of the targets, the target T's, in order to be able to do the subsequent task, which was a change detection test. And what happened in the change detection was that one of the targets could have changed the orientation. So they had to remember all of them to be able to do the task. And here is a sample display of the eye movements that we observed during our tasks. So as you see, yes, eye movements. And also sorry, what we found from some,
we couldn't hear you in the last 20 seconds.
Oh, sorry, I will just repeat that. So that we observed quite a few refixations in our eye movement data. And what we had also found from earlier studies is that there is a particular effect that the task relevance of an item has on eye movement behavior. So for example, it affects fixation durations. Fixation durations on targets are longer than distractors, which is kind of expected and understandable. But what we also found was an effect on refixation behavior, that more target refixations were made in correct trials and incorrect trials, and the opposite was found for distractor refixations. So what we thought was that target and distractor refixations are made for different reasons and are driven differently. Target refixations are made for rehearsal, which means that you have to go back and remember the orientation of the target and also know where the target is. Whereas distractor refixations are probably made because you've forgotten, and in the process of new search, all you have to remember is the distracted location so that you know you do not go back, which means that there is probably a difference in processing load or working memory load between targets and distractors. And this is what we expected to affect saccade planning. So that is the question we asked. So how does task-related refixation behavior affect saccade planning, which means for targets and distractors? And as I said, this was part of an effort to study EEG during such tasks. So we looked at the saccade-related potential, or SRPs, which is the EEG activity in the interval preceding a saccade. And what we did was we compared first fixations and refixations for targets and distractors. Now, of course, to do this, we had to co-register EEG and eye movements. And what happens essentially in this is that you can sync EEG activity to eye movement events, then you can see how does the brain respond to, let us say, what is there at gaze, or before it in our case, for example, because we're interested in pre-saccadic activity. Now, let us look at what happens in the brain during a saccade or around an eye movement. So here you see the saccadic spike activity, which happens at saccade onset. And here you see the lambda activity, which is the brain's response to the input at gaze. And here you have the pre-saccadic activity, which is the interval where we are planning for the next saccade, and that shift in attention to the new location this is where it happens. So this is the area we are interested in. Now here's the problem. When you talk about EEG and eye movements, the EEG is not only affected by the information at the fovea, but it's also affected by eye movement parameters. And in an earlier study, we found that the interval that we are interested in, the pre-saccadic activity, is affected by the preceding saccade size and the preceding fixation duration. Now, if these two parameters differ systematically between our experimental conditions, then we have a problem because we have a confound. So how do we solve this? One solution is to model the eye movement parameters as predictors of the EEG. We saw in two earlier talks and earlier sessions today, one by Benedict and one by Anne, that these can be modeled non-linearly. Another approach is just to select eye movements with that match in these parameters across experimental conditions to ensure that eye movement parameters do not differ between what you're comparing. And we chose this approach so that we will be able to compare our SRPs to traditional SRPs related to saccade planning that have been seen in literature. So now let's first just look at the eye movements. So as you see here, you have the first fixations have longer fixation durations than refixations, and this is what you would expect because perhaps they are attending longer, there is more working memory or more processing load in this, and this is all good. But this is exactly the kind of thing that can also affect EEG, and therefore we have to match these parameters, right? So this is how we did the SRP comparison. We put together all fixations and refixations, and we selected those that were matched for duration, saccade duration, and rank. Rank meaning where during the 10-second trial do these lie, so that we can pick up those epochs that are all happening at the same time points of processing during a trial. And then we selected 160 millisecond epochs before the onset of a saccade. And just remember that this is the tail end of a fixation on a target or a distractor, which means that we are trying to ask, how does the current item, which is the target or the distractor, affect subsequent saccade planning? We chose 8 regions of interest corresponding to the 1020 system of electrode placement. And so we averaged about 56 electrodes here, and then we computed average SRP amplitudes for first fixations and refixations. And this is what we found. So for target, what we found is that there were higher SRP amplitudes for first fixations and refixations in the occipital areas, and a reversal in polarity for the frontal areas. And this is very neat because this is similar to a negative parieto-occipital wave that has been found during saccade planning, which means that we seem to have found some kind of an analog under free viewing conditions for saccade planning processes. And just as we expected, there is a difference between first fixation and refixation, which may be because of the processing differences between the two. This is what happened for distractors. We did not find any difference between first fixations and refixations, meaning that there were no processing differences in between the two, meaning there was no effect on subsequent saccade planning. Because we had pretty much decimated all our electrode data, we had a dense array net. What we did was we also supplemented this average SRP analysis with some spatiotemporal clustering, cluster-based permutation tests. So essentially what happens is that you try to identify continuous points in space or electrode space and time, and you call this a cluster. And where continuous points where there's a difference between the two conditions that you're comparing, you call this a cluster. And now these clusters are then compared against a permutation distribution to identify significant clusters. And this is what we did, both for our targets and distractors. The advantage is, as I was saying, you can use all your electrode data. And here again, we found very similar results. We found 2 clusters, an occipital cluster and a frontal cluster. And as you see, the amplitude of first fixations was higher than refixations SRPs, and the opposite was seen in the frontal cluster. We did not find such an effect for distractors. So to summarize, we asked this question of, does saccade planning or preparation during fixation or refixation depend on past relevance? And the highlight was trying to do this under free viewing conditions. So we were able to study the interaction between top-down and bottom-up control of eye movements in EEG under free viewing conditions. And what we found was that higher SRP amplitudes for first fixations than refixations were seen only for targets, indicating a difference in saccade planning. And this difference can be attributed to the high processing costs or working memory for first fixations, which can in turn affect saccade planning, which means disengagement of attention from the current location and shift to the new location. And this can be expensive for first fixations. That will be all. Thank you all for listening. This paper has been published. There are more details about the complicated matching procedure that we used in the paper as well. I think Korea has probably shared the link.
It's in the chat.
Yeah, it's in the chat. Then I am open to questions.
Thank you, Radha. It's a beautiful study. If you have any questions for Radha, please ask them in the question and answer panel, and we are happy to answer them. Do you think, I mean, while we are waiting for your questions, a quick question for you. If it seems like your task is hard, at least for the design you showed, it's very tiny images, right? And TNL is very confusing as well. Is there a specific reason you made it that hard, or?
Yes, it was because one thing we wanted was that we wanted that participants fixate on the particular target to be able to discriminate it and do not see it peripherally, so that we can have more precise classification of what is a fixation on a distractor and what is a fixation on a target. They did pretty well though, about 75 to 80% performance though, in spite of that, how difficult the task was. Yeah.
Mohammed Abdel Haq is saying for subjects to have fun.
They did. Yeah, they did. Actually, we gave feedback, right? So at the end of every block, we told them saying you have 80% or 90% results. And given that it's an EEG and a difficult setup, people were motivated to do better and beat themselves from the previous block. So it was a bit gamified in some sense.
Yeah, that sounds good. Thank you very much. If there is no more questions, that's the end of our sessions today. And I would like to thank all of our speakers, Anna Sophie, Alex, and Radha, for their beautiful talks and works. And many thanks to our audience for being active and asking questions. I should also thank to Melvin for giving us a technical help, although you couldn't see him. He's just with us all the time. I hope you enjoyed the session and will enjoy the rest of the NeuroMatch conference and following sessions. Thank you very much, everybody.
Thank you.
Guys, in the case that Poe doesn't join us, I heard that the best idea is to start 15 minutes later to be on time. Okay.
Okay.
Yeah. Sorry for the change of. Sid, do you let the attendees comes in? Because I see 0 attendees right now.
I did start it, but I don't think anyone has joined yet.
Okay.
I'm also streaming on YouTube.
Yeah.
People have joined
Great.
in there.
So in the case that people are streaming us on YouTube, apparently we don't have the first speaker, Paul. Unfortunately, he didn't show up today. Sorry for the change of plan, but we are going to start at in 15 minutes, and our first speaker is Jules. We start in 15 minutes. I guess just for announcement, we're expecting to have power for this section, but apparently it didn't show up. We really apologize for that. We have to wait for 10 minutes that our first speaker, Jules, start talking. But up to that time, we don't have any plan. Thanks again for joining for this session. See you in 10 minutes. Again, the announcement, we haven't started yet. Our first speaker unfortunately didn't join us today. We have our first speaker for today, Jules, that is talking in 7 minutes. Feel free if you want to come back at 15. We will start in 1 minute. Okay, hello everyone. Sorry we had to start 15 minutes late today. I am Ruzbeh, your host today. I am a postdoc researcher at UPenn in Philadelphia, rainy Philadelphia. If you can hear the noise in my background, sorry for that. And I am very delighted to have two speakers talking about the synaptic plasticity and recurrent neural networks. I'm very much looking forward to the talk. Before starting the session, this is an interactive session, so people can ask a question in Q&A or chat, or engage in discussion, post links if it's interesting to them. And feel free to ask me to bring you on a stage also if you think you need the follow-up questions. Thank you again, and I want to go to the first speaker today. I mean, second speaker on the schedule, but first speaker for today. Jules is a PhD student and expected defense very soon. He's working at the Brain Institute in Paris and work on developments of methods of analysis to identify the biological determinants of behavior. He has a strong background in mathematics and computer science. And in his intro that he sent me, I'm quoting what said, I have spent my thesis painfully embarrassing cognitive neuroscience and experimental science. I'm pretty sure you didn't, and we are very much looking forward to your talk. His talk is about blaming blunder on the brain. Can indifferent choice be driven by range adaptation or synaptic plasticity? Thank you so much again, and we are ready to hear your talk.
Do you see my slide?
Yes.
Perfect. So thanks for having me. So this paper I've been made with Jean Donizeau at the Motivational Brain Behavior, and as you mentioned, in the Paris Brain Institute. So to start easily, the beginning point of this talk is the idea that physiological constraints shapes neural circuits and the way we process information, and ultimately shapes how we behave. A prime example of this would be the idea of efficient coding proposed by Barlow in 1961, which states that given that neurons have limited range to fire and to respond to an input, they should select which information to keep and which information to filter out. Evidence for this theory has been found in the fly's vision neuron by Laughlin, in monkey's value neuron by Kobayashi, and more recently in the bias of human decision by Polanyi. But this kind of physiological constraint is kind of hard to analyze because first, they are not instrumental at all to a given task, let's say a decision task. The brain would be better without such constraints, and in a way, and there are several biological scenarios behind them. There could be several ways to implement plasticity to account for efficient coding. So in this work, what I'm trying to do is to characterize such biological constraints and how they affect neural decision process. So I do this through the joint analysis of artificial neural network activity and bold signal. So I will train a neural network model on the behavior of subjects faced to a task, extract the activity of the units of these neurons, and build a similarity matrix comparing trial by trial the activity of each of these neurons. I'll do the same for the fMRI scan and compare the two. So in this, I'm following a very classical model-based approach. My model will be first, well, the INN I just told you about, with bounded activation, such as Gaussian or sigmoid function, and with two additional physiological constraints, which would be on one side Hebbian plasticity, which is simply the idea that if units co-activate, their connection will strengthen. And the second idea is range adaptation. It's a dynamical version of efficient coding, if you want. We simply state that when a unit's response to an input brings no information about the input, the firing function of the units will adapt to bring more information. I'm applying this approach to the experimental data of Botvinnik-Nezer 2019, which is a very simple task where participants are asked to either accept or reject mixed gamble, where there are 50% chance of winning a given amount and 15% chance of losing this amount. Classically, this kind of tasks are analyzed through a neuroeconomics model, which is, or just a behavioral model, let's say, which is just a logistic function applied on a linear combination of gain and loss, plus a given bias. And the hallmark of this approach is to define an indifference point where the model predicts that the subject will basically don't mind accepting or rejecting, but in fact, they still decide, but the model is completely blind to this part, to these inputs. First thing we show is that by using 6 types of network, a network with only Gaussians, only sigmoid, Gaussians with range adaptation, sigmoid with range adaptation, the same for Hebbian plasticity. All these networks still are completely deterministic, but manage to exceed the performance of the logistic model, and especially around this so-called indifference points. Then we use this model to look at the bold signal of the subject in a bunch of different regions, in the motor area, the visual area, the cingulate area, the orbitofrontal cortex, insula, amygdala, and striatum, extracted the similarity matrix and compares them to the similarity matrix of the network doing the same task as the subject and answering the same way. And what we found was that only the Hebbian network shown here managed to relate to brain activity and not range adaptation network and not logistic or static network. And we also devised a model comparison technique, a model selection technique, sorry, to statistically assess that such regions behave in a Hebbian way. Then we tried to see if we could go a bit further and explain behavioral difference between the subjects. So, and more precisely, the choice inconsistency of the subject. So typically the decision that goes against the logistic model prediction. So we built an Hebbianness score for each of these regions, which is simply the added value of using an Hebbian network versus a default static network to describe the similarity matrix, the geometry, if you want, of the representation in these regions. And we managed to do this. We managed to predict the choice inconsistency of the subject, and especially with a contrast between the left amygdala and the right striatum, and a contrast based on the Hebbianness of sigmoid model. So to wrap up, in this work, I've shown that biology constraint can be discriminated by the mean of an array-stay analysis. Such constrained artificial network explain away some incoherent choice without using the idea of noise or error. We can find traces of this constraint in the bold signal, And we can use this trace to understand behavioral differences. And last but not least, all this result has been replicated in the second group of this study. Thanks for your attention. I'll be happy to have questions.
Thank you so much, Jules. That was great. Now we are open to have a question and answer. If you want to ask a question, write it down in Q&A or chat. Thank you again. And I see that your paper also is available on BioCuff if somebody wants, everybody can check it online. I have a question before people start asking question. So what is the structure of this CNN that you are using? Like how you make it simple?
I'm not really using CNN. I'm using very simple shallow network, and which are composed of just two layers. One layer that receive the gain, one layer that receives the loss, and the first layer is split into two different parts to give a population coding of the inputs. And then there is a computational layer that actually does the combinations of those two layers.
So you have like a two-layer fully connected network, right?
Exactly.
I see, I see.
Well,
and yeah, go ahead.
Except for how the inputs enters the network.
Right. There are two inputs. Okay, and then to use the RDM here and compare it to the fMRI data set, the bold data set you showed, you use the same architecture to compare.
Yes, it's for all the 6 networks. The only thing that differs is either to use a Gaussian activation function on both layers or a sigmoid on both layers. And then you can also add an Hebbian modulation of this intermediate weight, of the weight between the two layers, or a dynamical range adaptation of the sensitivity parameter of this second layer.
Oh, so I saw that there is a parameter for Hebbian. Where is that parameter? That you are changing Hebbianness of that.
Yes, here's the Hebbianness is just a dynamic modulation of a static weight. So here you would have, this would be the effective weight used by the network.
Right.
Which is composed of a static classic part. And so here's the S10 just for a sigmoid to bound it between 0 and 1. And an activation part, an Hebbian activation part that will grow if the input and the output are similar. So this is a term that dynamically computes the covariance between an input neuron and the output of the either Gaussian or sigmoid.
Oh, that's interesting. And can you also go to the slide that you were showing the comparison when you change the Hebbianness?
I'm not sure. This one?
So you had the curve that you had the Hebbianness and
it's the behavioral one. Yes.
Right. Yeah, this one. The right one, right? So this shows that at the. So how do you explain this one? It just shows that some middle it goes high and then down.
So first, do
you have explanation for that? Mm-hmm.
Yes, the logistic part here. Let's start with the logistic curve. It's just the idea. It just reflects that around the indifference points, people either accept or reject a gamble with the same difference or weighted difference between gain and loss. So, and I interpret the fact that all curves look a bit the same. All curves show a bell-shaped form, if you want. To me, only reflects that this indifference point actually reflects the point where subject just don't know and either accept or reject. But what's striking to me here is that there is a quite significant increase or decrease of this inconsistency as soon as you consider a temporal dynamic and a nonlinear dynamic in the decision model.
Yeah, it's very interesting. I see that it's a bit consistent with different type of networks, like different sigmoid function or parameters that we have.
Yes, different kind of network. There is also a bit of trivial thing in this result because just simply because network have way more parameters than a logistic model.
Right.
Yet they have exactly the same inputs. Which can be discussed for the model with temporal dynamics, but it's all networks still provide a deterministic function from input to output, from gain and loss level to decisions.
Thank you so much, Jules. That was great talk, and I'm pretty sure that that amazed the cognitive neuroscience, and I wish you the best also for your PhD defense. And we are hearing from you more in future, definitely. Thank you again. And by that, let me wrap up and go to the last speaker for today.
Thanks for your question.
Thanks. So our last speaker is Huzi. He's currently a third-year PhD student. He's a double major in psychology and neuroscience at Indiana University, Bloomington. And he's working with Joshua Brown, Dr. Joshua Brown. His research is about developing plausible learning rules for recurrent neural networks, RNNs, and the nature of low-dimensional representation in RNNs. And today he's showing his latest result to us. Thank you again, Huzi, for joining us today. Can you share your screen to make sure that?
Yeah, okay.
Again, for the attendees, feel free to ask a question. You don't need to stay until the end of the talk. You can write it sooner in a way that I know how to manage the question and A. Thank you again, Huzi.
Okay. Wait a second. Okay, now it's okay. We can start.
I guess that's oh yeah, okay, good.
Okay. So our project is about replay-based fast and statistical sequence learning with recurrent neural networks. So basically the general question we're trying to attack in this project is how is sequence learning implemented in the brain? And more specifically, we're asking two separate questions. So the first is online gradient approximation the only approach for biological plausible sequence learning? So we know there are a lot of learning algorithms for RNNs that they are like online approximation of BBT, right? But also we're asking the second question, that is backpropagation through time a proper analogy for the sequence learning in the brain? So we know BBTT is notorious for its requirement of external storage of hidden unit activities. But here we argue that BBTT could be possible in the brain based on two key observations. So the first is that the replay, either forward or reverse replay in the cortex and famously in the hippocampus, is crucial for learning, right? And the second fact is that the reverse temporal propagation of hidden activity is essential for backpropagation through time. Based on these two observations, we proposed our approach, and that is the brain may approximate backpropagation through time with two separate recurrent neural networks instead of one. So in this approach, in the first step, the rewarded sensory sequences induce fast sequence memory in RN1, and then the reverse replay of RN1 serve as two purposes. The first purpose is that it leads to fast learning of forward replay of itself, and that is a one-shot learning. But also is capable to train a reversible recurrent neural network, the RN2, to perform the task, to learn the task gradually, and that is the statistical learning. So together, RN1 and 2 form a local variant of BBTT. Here we'll start with RN2 first. So RN2 is a reversible coupled recurrent neural network with competing projections. So unlike previously using models with just one group of neurons, here we have two groups of neurons. So neuron group A and B, and they are mutually connected with two pair of connections. So the intuition is simple. If the red connection is on average stronger than the blue projection, then the whole network will be driven to run in the clockwise manner. But if the blue projection is stronger than the red projection, then the whole network will run in anti-clockwise manner. And that is the intuition behind the reversible, behind the reversibility. And there are multiple choices for the activation function and the dynamic equation you can use for this neural group A and B. And here we just use the simplest one, the rate neural network model with two branches for projections. The competing or the balance between these two projections determine the running direction. And here is just, we sampled 10 neuron fine rates from a 200 neuron network. And as you can see in the reverse running phase, the higher rate is perfectly reconstructed with the fine rate in the forward running phase, but in the flipped order. And if you compute the angle between the neuron pattern vector between the forward and reverse running, the angle is always near zero as the cosine theta is near 1. So based on this architecture, the gradient propagation is plausible because we only require local information in the reverse replay phase. We can compute the history as we need. But this network cell itself is incapable to deal with inputs, because if you look at the computation graph, the unfolded temporal computation graph, you find that in the reverse replay phase, neuron group A and B here do not have knowledge about the inputs in previous time step. And because of this, we need another neural network to deal with inputs. And that comes the RN1. So RN1 is about faster reverse replay with also competing projections, but the architecture is slightly simpler. So this neural network, it receives two branches of projections originating from itself, attuned by the common gating coefficient gamma. So the gamma can be generated by some external oscillating signal or itself. These two combined projection capital F and capital G, they build a line and point attractors in the state space. Basically, it's a variant of the continuous time Hopfield network. So this network in the end combined arbitrary input patterns as a classical Hopfield network does, but in the temporal domain. So it learns different input patterns together and then associate them together and then play them back in a reversed order. This network only requires Hebbian learning rules, and in the reverse replay phase, it can do two kinds of things. So first, if we amplify the learning rates of these two weight matrices, then the whole network in the reverse replay phase will learn the reverse of the reverse replay, and that is the forward replay of the reverted sensory sequences. But also, you can just generate those reverse replay patterns and feed them into the RN2, and then the RN2 can learn the task finally. So this is the general workflow. So the animal will initially the animal will randomly explore the environment and generate some trajectories which lead to the reward. And the reward will trigger the learning in the first neural network. This RN can either learn the faster forward replay or can be used to generate patterns used to train the reverse replay in RN2. Then the RN2, based on our second slide, can perform the BPTT-like training and finally master the task. So we test this workflow with a simple T-maze navigation task with different Q type indicating different reward position either in the left or right end of different branch of this T-maze. And turns out that our model can successfully learn the task. So to the end, in this project, we propose a new type of sequence learning system of recurrent neural networks, which only requires only local information to perform gradient descent. And they bridge the gap between the observation hippocampus and brain, those replays, and also the long dilemma in the biological plausibility of BPTT. And yeah, basically that's all. So this, I was previously thinking that time was intense, so I basically ignore all the details. So if I have questions about that, feel free to ask.
No worries, you have time. but if you want to continue, you can continue. I can wait for 1-2 minutes to have a question.
Okay, so I have a few things I want to mention here in this slide. So for this neural network, the red projection, we can call them forward projections. So the red projection, they are just initially randomly initialized. So the whole network will randomly run in the forward running phase. But those blue projections, they will try to cancel these red projections, which makes the idea of detailed synaptic balance, right? And since this GA, so if we directly look at the equation, the task of this GA and GB, their target is to cancel this FA and FB in order to implement the anti-clockwise running. So if you consider the simplest case, the linear case, that is the forward running, they're just a linear transform. Then the backward matrix, they're also linear transform. And you can see here, there exists an analytic solution for these backward projections to cancel this forward projection. And that basically means the whole network will learn the reverse replay within very short time because just a linear regression. So that's how the reverse replay is implemented in this in RN2. Yeah, so that.
Thank you so much. Thank you so much, Huzi. I have one question by Jules that he asked: would it be possible to alter or lesion one RNN while letting the second one as it is? What would be your behavior prediction? That's an interesting question. So basically, if you do lesion study here for this architecture that you are working.
Okay, so basically this model matches some lesion studies. So for example, you can view this RN1 as a fast cache, which can be hippocampus, right? And the RN2 can be a general model of the cortex. So if you separate hippocampus and the cortex, you'll find that the whole network, the whole system is capable to perform like the fast learning, but it's impossible to consolidate those memories, transfer them into knowledge that can last for a long time, because the waves increase and decay in a very fast way in the first RNN, and that makes it impossible to learn those statistics in the task.
And if you do virtual lesion of your model and still stimulate it with the same input, would you see the kind of distortion predicted by the lesion study?
I haven't done that, but I'm pretty sure that if we just separate them, but the RN1 is still capable to learn a task. But if the task is changed a little bit, RN1 need to learn a task from beginning. So it has no knowledge about the statistics. It can just remember the trajectory.
So Huzi, for the training this network, you use a left and right paradigm, right? But how lengthy is your sequence that you're training?
Okay. Oh, so yeah. So this one is just for demo purpose. So in the real training, the time step is near 100. So we have near like 50 time steps for those for this branch and 50 time steps for this branch. And it can be actually extended to arbitrarily long as does not like jump longer than the threshold that BPTT can perform, because the limit of this algorithm, because since it simulates BPTT, is the performance.
Do you have a result on that? That
would
be interesting comparison. Do you have a result on that to compare that?
Oh, you mean the comparison between BPTT and this algorithm, right?
Yeah.
I showed I don't have a figure to show that, but you can for this figure, I done the experiment, but I didn't show a figure here. So the learning curve is pretty similar. So they have comparable performance. The only performance loss comes from the way we simulate BPTT, and I can show here. Yeah. So in the reverse replay phase, when we propagate the gradient in the reverse order, we need the transpose of weight matrices right here, and that's the problem that's the credit assignment problem in deep learning. And here we use the feedback aligned matrix, the random
So that's basically the performance bottleneck, and this prevents us to learn really long sequences. Yeah, that is interesting. Yes, thank you so much. I guess by that we have to wrap up this section. Thank you so much, Jules and Huzi, for joining us today. That was a great talk, and we are pretty sure that we hear more about your work in future. I hope to see you virtually again in the rest of the conference, and have a good rest of the day, everybody.
Bye, guys.
Bye.
Welcome. I hope to see you virtually again in the rest of the conference.
We're live.
I'm just going to allow a few more seconds so that people can join in. All right, I think we can get started right now. Welcome, everyone. My name is Shankar, and along with me, I have Nicola running today's session. I hope you're having a good time at NeuroMatch so far. Please let us know how you feel through Twitter, and when you do, please use the hashtag NMC3. In this session, we are excited to learn from our colleagues about brain networks underlying normal function and impairments in humans. The format of the current session is an interactive talk, which means the speakers have about 5 minutes to introduce their topic, following which we have about 10 minutes for question and answers. If you would like to ask a question yourself, we can bring you on screen. If not, you can always ask the question using the Q&A panel in Zoom. And also, please vote for questions that seem to be relevant to you. You can also chat with other attendees and panelists using the chat. And we just encourage people to be more positive and be free in discussing in the chat. So without further ado, we'll be bringing the speakers. First up, we have Charlotte Maschke from McGill University, who will be talking about how networks can be used to identify levels of consciousness. The floor is yours, Charlotte.
Okay, thank you. I will start by sharing my screen. Okay. So thank you so much for being here. Thank you for inviting me, or yeah, accepting me. So I would like to present my research, which is about dynamic functional connectivity for predicting the level of consciousness or even recovery of consciousness in behaviorally unresponsive patients. So when I'm talking about behaviorally unresponsive patients, I'm talking about patients in a disorder of consciousness. And for those who might not be familiar with this topic, I would like to introduce two short concepts, which are awareness and wakefulness. So having you as my audience, I expect like everyone to be in a normal conscious state. So every one of you should have a high awareness. So you are aware of what I'm saying and what happens around you. And you are probably wakeful. So you're awake and aware of what's happening around you. For patients in a coma, this looks completely different. Patients in a coma usually have a low awareness and a low wakefulness. So they are not awake and they're not aware of what's happening around them. Patients in a vegetative state usually have a high awareness, but a low wakefulness. So that means they are aware of what's happening around them, but they are not awake. From outside, you would feel like they are not awake, they're sleeping, they can't interact with the world around them. They can't react to, like, follow commands or react in any way. And the third part that I introduce is the minimally conscious state, where it is really difficult to assess awareness. So we just don't really know how they feel about how aware they are of their own environment. And their level of wakefulness is like constantly fluctuating over time. Sometimes they're responding like a little responsive, sometimes they're moving, sometimes they're not. So these three states, coma, vegetative state, and milli conscious state, are I'm referring to disorders of consciousness. And in my research, I'm working with this patient population. And I'm working with a patient population of 24 patients in a disorder of consciousness. And from these 24 patients, only 5 recovered consciousness in about 3 months after the assessment. 13 did not recover consciousness until today. And 6 of these patients did not recover consciousness, but shown signs of cognitive motor dissociation. And for those who don't know, cognitive motor dissociation means that they are able to command follow in their minds, so they can think about following commands. There's a really well-known study by Owen in 2016, where he showed if he's asking patients in a vegetative state to imagine playing tennis, then you can see in the MRI scanner that they are really imagining to playing tennis. So that means they are aware of the scientific around them having asked them to play tennis, and they followed the command, but they were not able to move their arm during this. So these are the disorders of consciousness patients, and I compare them to a healthy group, to a group of healthy controls where I have 9 adults. So the question in this research is, how can I predict who will recover consciousness from these patients? My methods are time-resolved functional connectivity. For those who are familiar, it's the WPLI, and I calculated from EEG. We are using 129 electrodes high-density EEG, and I'm calculating the functional connectivity in a time-resolved way. So when you look at these plots, you see the different brain regions, and the more red the line is, the more these brain regions are communicating together in one time step. And as this video shows, you see that this is a highly dynamic state and that the communication is really fluctuating over the whole brain. And this brain actually is a patient in a coma who later recovered consciousness and who wasn't responsive at all from the outside. So I'm calculating the time-resolved functional connectivity. And after this step, I'm performing some clustering methods. So I'm doing unsupervised machine learning, and I'm implementing K-means clustering in the hidden Markov model, where I go from the raw EEG data to the time-resolved function connectivity. And over here, I have a K-means clustering and a hidden Markov model. I identify different states within this time-resolved function connectivity. And I use these states to characterize one patient's characteristic, like how the functional connectivity evolves over time. My research for now has 3 main results, and I will go through them shortly and be free for some questions afterwards. So first, there is a high occurrence of low connectivity time steps in non-recovered patients. So here, the left-hand side, you see the K-means clustering result, and the more higher the bar is, the more often this cluster occurs in the time series of a patient. So you see here the non-recovered group in orange has much more time steps in this phase where the brain is really, really slightly connected, like not that much connected. That's almost why there's like really few connectivity. And the hidden Markov model identified the same state to be as the K-means clustering, mostly expressed in the non-recovered patients. However, here in blue, the recovered patients spend much less time in this phase, and the healthy adults almost spend no time in this phase. The same principle accounted for the so-called directed phase-lag index, where we don't just assess the strength of connectivity between 2 brain regions, but also the directionality of functional connectivity. So if this plot would be blue, that would mean that the brain mostly computes feed forward. So from the back, information flows to the front. If it would be more red, then it would mean feedback. So the information goes more from the front to the back. However, you see that these plots look completely green. So it's like undirected information flow, or it can't really be quantified. And the K-means clustering, as well as the hidden Markov model, found this state to be most expressed in non-recovered patients here in the orange group, as well as over there. And healthy adults, again, spend really few time, and even recovered patients spend fewer time than the non-recovered patients. Here in the hidden Markov model, almost at the level of healthy adults. And the third result is that I've identified some patterns like healthy pedals, which only occurred in healthy adults, but did not occur in patients with a disorder of consciousness. So here you see only the red bar, so healthy adults showed a high occurrence of these states. And these states were like really feedback connectivity in the dorsal path and some feed-forward connectivity in the ventral path, or the same in the inverse state. These plots refer to the upper plot, but this plot had the same characteristics in the bar plots. So these are my three results. And if I go to the summary, then I just have two statements. The first of all is the brain's functionality is based on a highly spatially and temporally differentiated activity. And if a patient is able to maintain this level of high dynamically fluctuation, then I suggest that he has a higher possibility to recover consciousness. So a maintained level of diverse and dynamic functional connectivity characterize a healthy brain and a brain which has the potential to recover consciousness. So these were my results, and I would like to thank the Biop Lab for their support, and Stephanie Blay-Moreas, who's my supervisor. I would be happy to take questions.
Thank you, Charlotte. So that's the question. That's the applause that you're getting on behalf of the attendees from me. So we have a question right now from Aldana. What measure of functional connectivity did you use? Is it Pearson correlation? Was the maximum functional connectivity equal to 0.5?
I'm using the weighted phase-lag index, which is a phase-based connectivity measure. So I'm measuring the phase coherence between two signals, between two electrodes. So this is the weighted phase-lag index. And for the directionality, I'm using the directed phase-lag index, which also starts from the phase coherence of two signals, but then goes further into which signal is phase leading and which signal is phase lagging, and then quantifies the direction of the information flow.
Great, thanks. As we are waiting for questions, I had a general question about how you define awareness versus wakefulness. So how do people know if a person is aware if they are not able to move their eyes or any kind of motor movement?
Yes, that's a really great question, and I went through this super quickly, but it's a super important concept that I didn't want to spend too much time because it's not my research topic. But Adrian Owen in 2006 was able to, he put a patient in a vegetative state into an MRI scanner, and the patient from the outside was completely unresponsive and he didn't follow any command, and people from the outside didn't know whether he understands, whether he's aware of people around them, whether he's able to understand human language, or whether there's any kind of awareness. So it's super hard to evaluate awareness from the outside. But he put them in a scanner and asked them to imagine to play tennis, to just move around the right arm and play tennis. And when he looked at the scanner, after he has asked the question, there was the left-hand side, the motor area, flashing up and taking a lot of oxygen. So he knew in this time that the patient actively understood his question, was able to follow the command of imagination of playing tennis. And the other task was spatial navigation. So after playing tennis, he said, okay, thank you. Now stop to imagine playing tennis. Now start to imagine to navigate around your house. And so the motor area stopped being active in the scan. And suddenly the more spatial navigation related areas started to flash up. And this is all the research he has done around it. So there's a long story of more further research about this, which validated it. But with these methods, he's able to evaluate whether patients who aren't responsive in the behavioral way are still able to respond with just their thoughts, with just reading their brain activity.
Oh, that's really cool. Do you know if there are any other like EEG-based studies that do this, like able to differentiate?
The lab was working on further measures because suddenly there was a high interest of many people asking, because they had patients in which they stayed during a coma at home, they were taken care of, and caregivers started asking, how can I get this measure for my relative I take care of? And it was often hard to move them to hospital to have a MRI unit, and it's so expensive to do this and so hard to organize it. So they have evaluated the same paradigm for an EEG scan, and they're actually they afford a car with the whole EEG equipment, which is portable, and they go to a person's home and they're testing
That's cool.
these tests. So Adrian Owen, if you're more interested in this, go to his lab's website and you'll find many information about this.
Okay, perfect. So if we don't have any more questions, I have one more. When you mean in your second result, when you say it's undirected, is it undirected or is it like bidirectional coupling?
It can be both, but I think we would need to interpret it in the light of the first result, because I think these two states are connected. It's like states where the activity is super low and there can't be identified a pattern of directionality. And this might be either because it's like undirected or because there's just no information flow at all.
Oh, got it.
So these measures are really connected one to each other.
Okay. All right. Okay. Sure.
Can I ask a question?
Yes, go ahead.
So Charlotte, it was fascinating talk. Congratulations on that.
Thank you.
I wanted to know, because there are many individual differences, especially the non-recovered patients, then I was wondering what other measures had you taken into account? Like, because we know that there are like differences in abilities, like, for example, I could think of brain age, or I don't know, attentional differences or things like this. What have you considered, like, to look within this group of non-recovered patients?
Yes, that's also a really, really good question. And that's a hard point. When we do this research, it's hard to get our participants, and they are most of the time brain injured. And most of the time it's so hard to find a common group of patients. So we evaluate their brain injury. We know sometimes we only analyze one hemisphere. For example, if one hemisphere is completely damaged, then we tend to do this. But in this study, I take into account the time since the injury. I take into account the age of the participant, as well as gender difference. But yes. So I'm trying to have the groups as balanced as possible, but especially also for the recovered group where only 5 patients recovered, it's really hard to account for all the individual difference. So this would be a part which need to be discussed in the discussion.
Right.
But it's still promising to find a measure where all individuals, even if they have their different brain damage and their different characteristics, all have like the same spot in a plot and the same characteristics, which makes them different from all other patients, and it's still compelling.
Right, absolutely. Thank you.
Right.
But it's definitely a problem.
Yeah. Let's thank Charlotte again. And next up, we have Adriana Ruiz-Rizzo from Ludwig Maximilian University of Munich. She'll be talking about networks underlying visual processing. The floor is yours, Adriana.
All right, thank you very much. So I'm going to talk about functional connectivity between right frontoparietal and visual networks that are linked to visual processing speed. Oops. Yeah, so visual processing speed is a visual attention function that represents how fast we process visual information. And as you can imagine, it is important for many daily activities that rely on visual information, like making decisions, encoding information when reading a book, or navigating through the streets of a city. We can measure visual processing speed in the lab by using a whole report task, where we present stimuli, like you can see here, a group of letters, and then we ask participants to report all the letters they are fairly certain they have seen. And that's why we rely on accuracy for performance instead of motor speed or reaction time. What we do instead is varying exposure durations, and we do that to estimate visual processing speed by using an exponential growth function like the one you see here. And we expect that by increasing exposure duration, the accuracy of report would also increase. We quantify visual processing speed with this parameter here, parameter C, as the slope of the curve at its origin. And it quantifies the number of letters that can be represented in a second. Now, if we want to study the neural correlates of visual processing speed, we can use functional connectivity that is based on resting state functional magnetic resonance imaging that uses the fluctuations in the blood oxygenation level dependent signal, or BOLD signal in short. And by using diverse methods like independent component analysis, we can obtain different sets of brain regions that we can name as brain networks, and each one of them will have a characteristic or specific time course of these BOLD signal fluctuations. Previous studies that have used these approaches have identified networks that are relevant for visual processing speed. More specifically, the connectivity within so-called singulo-percular network and the between network connectivity of the singulo-percular with the right frontoparietal network have been described as relevant for visual processing speed. However, from a theoretical perspective, but also from a practical point of view, the visual regions should also be involved. And that's why in this study, we wanted to investigate whether these higher order singulo-percular and right frontoparietal networks are related to visual regions for supporting visual processing speed. So this is what we did in this study. We assessed a sample of 48 healthy participants, and they underwent resting state functional magnetic resonance imaging. And importantly for this study, we analyzed the entire frequency range available in our data, and that was determined by the sampling rate. We did that in separate frequency bins from the slowest, or slow 5, to the fastest, or slow 2. And as I said earlier, we analyzed the between network functional connectivity of the singulo-percular, right frontoparietal, and visual networks. And we examined their association with visual processing speed. So here are the results. And here I want to give a shout out to Svenja Kuchenhoff, a brilliant master's student who did most of this work. So we first identified the singulo-percular and right frontoparietal networks across frequency bins, as you see here, and also the same for the visual networks. And we identified 5 different visual networks across the different frequency bins. Next, we looked at the between network correlations or connectivity. And this is what you see here in these different matrices that each one represents one frequency bin. In blue is represented the negative connectivity, whereas in red colors, it's the positive connectivity. What you should care about here the most is that the across bins, the connectivity between singulo-percular and right frontoparietal network was mostly negative. There was one exception here for this. This is a subcomponent of the singulo-percular network where it was mostly positive. Next, we looked whether there were differences across bins for this between network connectivity. And here we're plotting the connectivity of the singulo-percular network with visual networks across bins. And as you can see, it was strongest and positive for the slow 3 bin in the insula subcomponent. This slow 3 is the second fastest bin that we analyzed. Next, for the right frontoparietal and visual networks, we found that it was strongest and negative in the slowest frequency bins, so slow 5 and slow 4. Next, our main question, of course, was to see the association with visual processing speed. And to do that, we ran 5 linear regression models, one that included each of the 5 visual networks that we found, and we included as predictors the connectivity with the singular opercular network and with the right frontoparietal networks. I'm going to show first predictors for the singular opercular network. So here we have the different predictors across the different frequency bins and also represented in each color for each different linear regression model. And what we should notice here is that all of these bars are crossing the midline. And this is representing here the estimates of the predictors along with their confidence, 95% confidence interval. And because all of them are crossing the midline, then we can say that none of these predictors significantly predicted visual processing speed. In contrast, for the right frontoparietal network, we can see that in 2 models involving these 2 different visual networks, there were 2 predictors in slow 5 and in slow 2 that did not cross this midline, indicating that they significantly predicted visual processing speed. In sum, we found that functional connectivity with visual networks was strongest and positive in slow 3, so the second fastest bin for the singular opercular network. And this was specific to the insula subcomponent. And for the right frontoparietal network, in contrast, it was strongest and negative in the slowest bins, slow 5 and slow 4. We also found that the right frontoparietal, but not the singular opercular network connectivity with visual networks was associated with visual processing speed, and this was observed both for slow 5 and slow 2. So our results, in light of the previous evidence, suggest that the right frontoparietal network could be the interface between the computations generated in the singular opercular network and there are later implemented in visual networks to support visual processing speed. And with this, I'd like to end my presentation and thank the co-authors of this work, especially Svenja, who did most of the work, the NeuroMatch conference organizers, the funding agencies, and you for your attention. And I'm happy to take questions.
Thanks for the wonderful talk, Adriana. So we have a question from Hanwin. Did you try to compare ICA to use Atlas to define the brain network?
Yes, that's exactly how we define the networks. So we use the templates of Allen and colleagues published in 2011 in Frontiers in Human Neuroscience, I believe. So we did 75 decomposition and we did these different ICAs for each frequency bin. And we compared our networks or our components with these templates to define the networks of interest.
All right. Next, we have a question from Quan Wan. What is the significance of the different frequency bins here? Like, why do you separate them into these frequency bins?
Yeah, so we follow that approach because previous studies that have capitalized on the frequency information have found that there is meaningful information around the highest or the upper limit of for filtering the data, the resting state fMRI data. So that's why we chose to encompass all the frequencies that we could have given our TR. And the other thing is that, so about the significance of the different frequency bins, we think, I mean, if we look at the history of resting state fMRI, it was initially thought that it was just noise in the signal. It was usually discarded and avoided. And that's why we have to fine tune how we analyze these data. And that's why we still need to better understand the temporal or the frequency information contained in these data. So I don't know, I mean, of course, it is difficult to say that these networks are just communicating in this frequency in the same way that we see the oscillations with EEG. We cannot say that just the networks prefer this specific frequency range, but we could say that we can fine tune the analysis of resting state fMRI to find these meaningful correlations with behavior. This is at least how we see it so far.
Okay, great. So we have a question from Conrad Cording. What do the results mean about computation in the brain?
But in terms of visual processing speed specifically, or in terms of what the frequency bins mean?
I hope he can clarify this, but I guess it's mostly in terms of, in general, like with this connectivity, how does this help in terms of understanding computation?
Right. So all our study and our methods are based on the neural interpretation of the theory of visual attention, and it proposes a mathematical model to understanding how visual attention works and how these the computations that are like make possible visual attention. And so one of the mechanisms that it's proposed is it's called pigeonholing, which means that there is an increase in the firing rate of neurons that code specific categories. So we can have attention directed to specific objects, but we can also have attention directed to specific categories. In the case of when we assess visual processing speed with this whole report task, we're not asking the subject to focus on objects. We're asking the subject to focus on categories. So in this case, letter categories. So the attention is not directed to filtering out specific objects that do not fulfill the criterion proposed in the task, but rather just the category of letter in general. So what is proposed is that every time we are focusing our attention on selecting categories, then there is an increase in the firing rate of neurons in the visual cortex that code for that category. So that would be in terms of the computation. Of course, this is theoretical still.
Okay. And so we have one more question from EC Zainab. Great work, thank you. I wonder, did you check the hand orientation of participants? Would you expect different hemispheric results due to this?
That's a good question, because although we are measuring in general visual processing speed, the stimuli we're using is it's verbal stimuli, because those are letters. I wouldn't be able to say whether there would be a difference for like depending on the hand dominance, but what is known at least is that the right frontoparietal network is important for alertness function, for phasic alertness, for example. And this seems independent of the hand dominance of participants. So I wouldn't expect at least a difference depending on the hand dominance of participants for that network.
Okay. I think we are, yeah, that's about the questions. I think we are running on time. Thanks for your talk, Adriana. Sure. So we'll be moving on to our final talk today. It's by Fateme Akrami, who will be talking about network differences that we observe in patients with bipolar disorder. Fatema, it's your turn.
Okay, thank you. In this study, we want to investigate about bipolar disorder and its alteration on topology and synchronizability of functional brain network. It's shown that bipolar disorder impact on cognitive functions such as speech, behavior, and decision-making. And we know that some deficit in brain regions associated with bipolar disorder and functional brain network impairment is related to disorder as well. On the other hand, graph theory provides an opportunity for us to identify the connectivity between multiple brain regions, and we use it to know about the bipolar impact on brain. Previous study showed that in this study, they used magnetic resonance imaging, and they found that in BD, among BD participants, we have longer characteristic path lengths, but clustering coefficient decrease, as well as global efficiency relative. On the other study, they use EEG and graph theory analysis show that synchronization in alpha band and clustering coefficient, as well as global efficiency decrease, but clustering path lengths increase among BD individuals.
We know that bipolar disorder changes brain waves activity and powers. For example, there are increase in theta activity, and synchronization is decreased in alpha band, and in the resting state eye closed condition. So there was a question for us that what happened in the brain in BD condition in the resting state eye open. We focus on the brain synchronizability in theta wavelengths in eye open condition, and we know that theta wavelengths is more related to memory, navigation, and brain-wide network processing. We have 21 BD and 21 healthy participants. We use EEG for data collecting, and for pre-processing, we use EEG lab, and we use Loretta for localization, and ultimately graph theory for analysis. Our results show that there are significant difference in theta band in clustering coefficient. You can see that among BD participants, we have increase in clustering coefficient and increase in global efficiency, but decrease in the smallest, second smallest eigenvalue. As mentioned before, in this study, we use graph theoretical analysis to investigate topological among BD patients. We use EEG as well, and we found out in theta band, clustering coefficient increase. It means that segregation and local processing increase among these patients, and global efficiency increase as well, which more related to integration and transferring data. But second smallest eigenvalue decrease, which show that robustness and resistance in network decrease. So our findings support the hypothesis that in theta band, the functional topological of brain network in eye open condition is affected by BD. In conclusion, we can say that BD as a disorder changes brain synchronizability and alters its topology, and in theta band, brain wave activity change, so data transferring and data processing influence on this disorder. Thank you.
Thanks for the wonderful talk, Fateme. As we are waiting for people to type in their questions, I had a quick question about how you build these graphs. So what type of measure did you use to calculate the connectivity?
We use lag coherence.
Oh, lag coherence, phase lag coherence. Okay, got it. Did you see any other any differences in other frequency bands like alpha band, because that's the most strongest band when you have an resting
Not
state, right? Not significant.
In eye open condition, no.
Okay, got it. So yeah, there is one more question from Cornet Coding. How do you interpret global efficiency?
Global efficiency is more related to, as I said, it's related to integration. This means that the integration in brain network increase, and it's more related to transferring the data. It means that the data transferring in theta band increase in BD condition.
So there is an increase in data transfer.
Yes.
Okay. That's so like, how do you interpret then what BD is actually, right? So we have these, I mean, these are people who have both like the manic and depressive episodes. Do you think this increased transfer is present throughout, or is it in only like certain phases of their disorder?
Sorry, I don't understand your question. Please repeat it again.
Sure. So in bipolar disorder,
so
BD, so in these patients, they exhibit a different symptoms at different time points, right? So at some points they are manic, and sometimes they are depressive.
Yes.
So do you think this global efficiency is increased throughout the entire period, or did you have any information on what phase of the disorder they were in when you collected the data?
Yeah, there are some differences between the people who suffer from bipolar disorder and depression in theta band. We select these people, and then we found out, yes, in among the people who have different theta band, the transferring data increase.
Okay, got it. Okay, so as we are looking for questions, if the other panelists have any questions, please feel free to unmute yourself and ask.
I think I have some more technical question about the graph theory. Are you thresholding the graph in any way, or are you taking the raw graph as it is? Because there's so many like thresholding methods to simplify the graph or binarizing it.
No.
You're taking the raw graph. Okay.
Yes.
And the second question is about the smallest eigenvalue.
Mm-hmm.
What is the motivation behind this? I'm not familiar with the details of this graph theory analysis, but how do you come from the eigenvalue to the robustness of the network?
Let me ask my co-author to answer this question. Sorry.
Oh, he's there. Great.
Hi.
Hello.
Hello. So the second smallest eigenvalue is from the spectral graph theory, and a spectral graph theory technically talk about the dynamic of the graph. And first we need to calculate all of the eigenvalues and eigenvectors of the adjacency matrix, and after that we can see the spectrum of the eigenvalues. And especially second smallest eigenvalue is related to the dynamic of the graph, the robustness of the dynamic of the graph. So when we have higher second smallest eigenvalue, we can say the graph is more is in a more robustness condition, and when we have lower value, then the robustness is decreased. And we saw that in the patients, the robustness has been decreased. Yeah.
Thank you. That's really comprehensive answer.
Sorry.
I know it's a really technical question. I'm super interested in this. Thank you.
Yeah.
All right. So if we don't have any more questions, I think we can probably wrap up the session. And I'd like to thank the panelists again for these wonderful talks, and also the attendees for being here. And just a quick reminder, please feel free to share your information about the conference in social media networks. And when you do, please use the hashtag NMC3. Otherwise, thank you and hope everyone has a good day. Bye-bye.
Thank you, Shankar.
Thank you.
Thank you.